Hello, everyone, and thank you for joining us for day 2 of the Seattle Cell Science Symposium. Good morning to our West Coast colleagues, happy lunchtime to our East Coast friends, and good evening to those joining us from Europe and elsewhere. We are excited to have you here. We are just going to go through a few housekeeping remarks before we get started today, right at the half past the hour. So first of all, we want to know who's here with us today. Please take a moment to introduce yourself in the chat. Please share your name, your affiliation, or where you're calling in from today, and let us know why you registered or what you're excited to learn about today. Be sure to send your chats to all panelists and attendees as noted on the slide. Welcome, Kathy from the Frontiers Group from Seattle. Again, if you're just joining us, please feel free to introduce yourself in the chat window. Be sure to send your message to all panelists and attendees. Hello, welcome back, David from University of Arizona. Martin from UCSF, hello, good morning. Well, as you all continue to introduce yourselves, we have just a couple more housekeeping notes to share. When we get started, please be sure to share any questions you have for our presenters in the Q&A panel, which is accessible in your Zoom toolbar. Once you open the Q&A panel, you can click to type your question in the window that pops up. We ask that you begin the question with the speaker's name so we know to whom it's directed, and be sure to click send. If you are having any connectivity issues today, we have two options for you. The first is that you can view this event on our YouTube live stream by clicking the link that we are adding in the chat panel now. You are also welcome to call in from your phone for better audio by clicking the switch to phone audio option and following the prompts to dial in. This is available in the audio settings panel. Here is a brief overview of today's agenda. We will have some introductory remarks, followed by presentations and concluding with a panel discussion with all of our presenters. We do hope that you can stay for the entire program, but if you do have to step away, we will be posting a full recording of today's program on the event website and on our YouTube channel after the event. As a reminder, today is day 2 of the symposium. Yesterday's presentations were focused on cell nucleus research. If you were unable to join us, the webinar is available on our YouTube channel. Today, you'll be hearing 4 lightning talks from Allen Institute for Cell Science researchers, each highlighting a different resource available on our data portal, allencell.org. These lightning talks are meant to give an overview of the tools and their applications. We will be hosting a Q&A session for these 4 featured tools after the webinar concludes at 12:15 p.m. Pacific time. This is a separate Zoom meeting, so we ask that you check the event website. You can still register for this separate Q&A session. We are sending the link in the chat shortly, and we'll be sending it throughout the day. I'm going to take a couple moments, read out some more of our introductions in the chat. We have Karina from University of Washington, Tacoma. Welcome. Wayne from Seattle. Hey, neighbor. Christina from Evotech Biologics in Seattle as well. Hey, Tim Murphy-Stevens from the Allen Institute for Cell Science. Again, to learn more about the tools that we'll be featuring today, you can go to allencell.org. We'll also be hosting that question and answer session at the end of the end of this program, we have that separate meeting. The link to register is now in the chat. For those of you just joining us, please feel free to introduce yourself in that chat panel, send your name and your affiliation, where you're calling in from today. If you do have questions for our presenters, we highly encourage you to ask those questions in the Q&A panel, not the chat panel. Just need to open up that window. You can start typing your question. Please begin the question with the speaker's name and then hit send. Most of our presentations we have saved Q&A time for after, so we will be doing live Q&A and answering your questions. And if you are having any bandwidth issues, you can tune into our live stream on YouTube. If you're having Zoom connectivity issues, we'll send that link in the chat again, or you can switch to phone audio to minimize the strain on your bandwidth by finding the audio settings panel and clicking switch to phone audio and following those prompts to dial in. Just before we get started, we'll highlight the Day 2 agenda today. Start with opening remarks, followed by a series of presentations, and then a panel discussion with all presenters. And again, we'll be highlighting the tools Q&A session throughout the day. We have our 4 lightning talks just before the break today. And please be sure if you want to ask more questions about the tools, you can sign up for the Q&A session today. We are now at 8:30 a.m. Pacific time, and we are going to get started. And I'm happy to introduce Ru Gunawardene, the new Executive Director of the Allen Institute for Cell Science. Welcome, Ru.
Thank you, Megan. Welcome back, everyone. For those of you who attended yesterday and for those of you who are attending for the first time, welcome to Day 2 of our virtual Seattle Cell Science Symposium. So my name is Ru Gunawardene, and as Megan just said, I'm the new Executive Director and very excited to lead this amazing team. So today we have a really exciting lineup of speakers from kind of a range of topics in cell biology. So we focused on the nucleus yesterday, and today we are broadening the scope a little bit. And we also have a few people from the Institute who are going to highlight some of our own research and some of the tools that Megan just talked about. So exciting lineup. But I would like to start us off by, sorry, my computer's acting up, giving you a very brief introduction to the Institute, some of which you heard before from Susan yesterday, but worthwhile echoing some of this for the people who are attending for the first time today. So let me get going on that. So one of the main goals of our institute is to create this multidimensional map of cell states so that we can better understand the complex cell behaviors, including things like mitosis, differentiation, and various perturbations that the cells undergo. And our approach to do this is with imaging, because we really do believe images provide a rich source of information for us to really understand cell behavior. And to do this, we took this approach of kind of focusing on the major structures of the cell and doing live high-resolution 3D imaging, because we really believe that the organization of the cell can tell us a lot about what the cell is doing. And so this also allowed us to reduce the complexity of our measurements as well as the interpretation, rather than looking at all the genes or the proteins. It's kind of a dimension reduction approach. And what we hope to gain from this kind of analysis is to better define cell states kind of in a more holistic and multidimensional way, learn about the rules of the cell in terms of principles of morphogenesis, how do cells transition from one state to another or one cell type to another, and eventually with all these computational tools we are all using, be able to look at a cell and predict what it might do in the future. So we want to start this by looking at as normal a cell as possible, but of course we want to build on this by looking at other states, including disease and other pathological states. So for that reason, we have decided to use the iPSCs. Many advantages to this. One is it's as normal as we can get and still grow cells in the lab. So they're diploid, they're human, they're non-transformed, which is a big advantage over other cell types. And maybe the biggest advantage, though, is their pluripotency, which allows us to explore cell states all the way from the more epithelial-like stem pluripotent state through different lineages and cell types. So what we've done is to create a collection of isogenic lines using a normal healthy iPSC background so that we can do all this live imaging and analysis that you're going to hear about today and you heard a little bit about yesterday. So these ideas and concepts and the methods and the data are described in 2 recent manuscripts we've posted on bioRxiv. And if someone could post those links in the chat box, might be an easy access for our participants. So in the first paper, we focused on the pluripotent stem cell and trying to understand the integrated cell organization and the variance around it. So this is kind of our baseline to understand the rules of the cell. And this is a visualization of the many structures within the cell. So the different colors are different structures. And I'm just going to show you a movie so you can kind of see the 3D visualization of this. So I highly suggest going and taking a look at this paper because there's a lot of cool information in there. And the second paper, which we posted a few months back, talks about this concept of different data types to help better understand cell states. And here our approach was again to look at the organizational states, the structural states of the sarcomere in cardiomyocytes, and then ask whether looking at gene expression in the same cell, so integrating 2 types of readouts, could tell us more about what the cell is doing. And to get the answer, I would suggest reading the paper, but pretty much the bottom line is different types of data can provide information that's actually very, very useful to understand the various cell states that even one population can have. So to do all this and more, what we plan to do now and in the next few years is to take those concepts that I just described to you before and the methodologies and the tools we've built to explore cell states through differentiation. So we can do all this. So for now, we are going to focus on the mesoderm lineage because we have some experience imaging and studying the iPSC state as well as the cardiomyocytes. So we are looking forward to exploring the cell states between, including things like the early, the epithelial to mesenchymal transition that happens. So the idea again is the same goals as I mentioned a couple of slides ago, but to add the 4th dimension of time so that we study dynamics and also look at the genomic architecture as we take a deeper dive into the nucleus. So all of this work is done with team science as one of the drivers of our institute. So I have to thank the whole team, about 70 people. And all these people also helped us build this amazing set of tools that we developed kind of from scratch to image, to analyze and interpret some of this data that are also highlighted in those 2 papers. So you're going to hear about 4 of these tools today in our fun lightning talks later this morning. And one of our scientists will present on the iPSC state. But I also want to, before I end, thank some of the nonprofits who have helped us distribute these lines and these tools to the community, such as Coriell, who distributes our cell lines, and Agin, who distributes our plasmids. And I think during these challenging times, we need to support them as well, because without them, we couldn't do the kind of collaborative science we all want to do in the future. So with that, I would like to thank our founder for his amazing generosity, support, and encouragement, without which we wouldn't be here today. And with that, I want to thank you for again participating today. Looking forward to our exciting lineup of talks. And I would like to hand this off to Irina to introduce our first speaker.
Thank you, Ru. I'm Irina Müller, one of the scientific program managers at the Allen Institute for Cell Science, and I'm a member of the Symposium Planning Committee. I'm really happy to introduce our first speaker of the day, Dr. Susan Parkhurst. Dr. Parkhurst is a professor in the Basic Science Division at the Fred Hutchinson Cancer Research Center and an affiliate professor in the Department of Biology at the University of Washington. Her lab uses multidisciplinary approaches to study dynamic membrane cortical cytoskeleton interactions in normal cells and the consequences, what happens when these regulations go awry. Currently, the Parkhurst Lab is investigating these interactions in the context of wound repair and nuclear organization. Today, her talk will be focusing on the cell wound repair aspect and is titled Cell Wound Repair, Dealing with Life's Daily Trauma. Please welcome Susan.
Good morning, everybody. I'd like to start by thanking the Allen Institute and the organizers of the symposium for the opportunity to share some of our work on wound repair. So wounding occurs in response to many different things, including daily wear and tear, accidents and trauma, environmental stresses, and then clinical interventions and diseases. And this can happen at many different levels. So from single cells to tissues to organs, and an organism has to be able to repair these breaches very quickly in order to prevent death. My lab works on different aspects of wound repair, and I'm going to share today our work on single cell repair. So single cells undergo daily wear and tear and stresses from physiological and environmental assaults. And what they have to do is to repair their membrane breaches very quickly. So for example, if you walk down a hill, many of your muscle cells are being ripped open, and you have to be able to repair them because you can't replace all of them. And there have been a number of different models that have been set up to look at wound repair. They all function very similarly, and they gave us a sort of cellular events that are happening. And that's shown here. So this is the membrane in the cortical cytoskeleton. And when you have a membrane breach, what happens is calcium flows in. It redirects the actin and microtubule cytoskeleton towards the wound, and they bring vesicles to the surface. And those vesicles can fuse with each other and with the plasma membrane to plug the hole. And they do this within seconds so that nothing can flow in or out. Then an actin myosin ring forms on either side, around the periphery of the wound, and it will translocate inward to close the wound, both the membrane and the cortical cytoskeleton. And once you've closed the wound, you have to then remodel all of this actin and microtubules, cytoskeleton in the membrane in order to get it back to its original state in terms of composition and to restore its function. So in our case, we're using the Drosophila embryo, and there's a fluke of Drosophila development where the very early stages, you have nuclear divisions without cell divisions. So you have what is essentially a giant cell. And what we can do is wound and look just at the cortex right here. So this just shows you we have an embryo. It's encased in a vitellin membrane, which is an impermeant membrane. So it's really an isolated cell. We wound right on the lateral surface. And in this case, if I just draw a line through here and show you a cross section, here's the embryo. We put it onto a cover slip with glue, and we're imaging from the bottom. And the kind of thing we see is this. This is just a time-lapse movie using an actin reporter. And we can see what happens in an XY view and also what happens if we make orthogonal views. So we can see what's happening below the surface as well as what's on the surface. And what you'll notice is that there's an actin ring that's formed right after it's wounded, and that will translocate inward to close the wound. And this whole process takes about 30 minutes. So what we end up with is a wound that has, for example, a very high amount of actin right at the periphery that we call an actin ring, and accumulation of actin that's not quite so high that we call a halo. And in order to look at this, we do time-lapse microscopy, but that's hard to compare movie to movie. So what we do is to take a strip right down the middle of this image and then line up the time points this way. So you can see, for example, the actin ring closing. So wounds, when they first happen, will expand, then they'll contract and close. And eventually when they close, they need to disassemble that actin ring and then remodel all the cortex. And as I said, this whole process takes less than 30 minutes. So with Drosophila entering the model systems for single cell repair, that gave us a genetic system for the first time. And so we've been able to use this to look at individual molecules and individual events, but we can also take advantage of the fact that it's genetic and do screens. And I'm going to tell you a little bit about two different screens that we've done. looking for different aspects of wound repair. The first one is a microarray screen, and this is to look for genes that are up and down regulated in response to wounding. And the second one we call a fly trap screen, and that's to look at GFP tagged proteins and ask which ones are recruited or depleted upon wounding. And I'll start with the microarray one. And in this case, one of our, the reasons for doing this was to look for start signals. So we don't know what actually triggers wounds. Once it happens, how do you know you're wounded? And we were following sort of the prevailing model in the field, which said that it's an activation of transcription. And so that was a little bit at odds with our system, because our system is an enclosed system, and almost everything that's happening in an early embryo is coming from the mother, right? The mother has deposited RNAs and proteins, and that's running all of the really early events. And there's very little transcription. So if you need a transcriptional response, then we were wondering how that happened in our system, since it seemed to model all of the same things as other systems for single cell repair. Okay, so the way we did this was to take an embryo. We wounded it 8 times, and that was in order to get a robust enough response so that we'd be able to see up and down regulation. And that takes about 1,000 embryos, or ones that were not wounded. And then we would isolate RNA, make cDNA and label it, and then put it on a microarray. The particular chips that we used had 12,000 cDNAs on it, which represents about 60% of the fly genome. And we did 2 different time points. We did it right after we wounded, or as close to it as we could get, which is about 5 minutes, and then 30 minutes, which was at the end of the repair process. So the first one was hopefully to get anything that was a transcriptional start signal, if there are them. And then the 30-minute was to see anything that's involved in wound repair. And the idea was to identify these and then to use RNAi to knock them down and ask if they had a role in wound repair. Okay, so what we got, these are volcano plots showing the early time point and the late time point. And the black dots are the genes that don't change. And the red ones are ones that go down, green ones are ones that go up. And what you'll notice automatically is that in this early time point, we got no response. There are no genes that go up or go down. And so that fits very well with what we think happens in an early Drosophila embryo, where there's very little transcription. But we do see that transcription is involved if we look at the later steps. We wanted to look at this from a different aspect and see if it really is the case that you're not using transcription as a start signal. And so what we did was to inject an inhibitor of transcription. And so what I'm showing you here are wounds that are generated with an actin reporter in the background. So these are just 4 time points here, and then the chymograph, which shows the healing over time. And this is a buffer injected embryo, and this is very similar to what you would see without, just a wild type embryo. If we inject alpha-mannitine, which is a transcription inhibitor, the early steps look very similar to the buffer control. There are some differences though. The amount of actin that you accumulate is a little bit less. It's delayed in removing the actin's ring at the very end. So there are differences, and it looks like transcription is involved, but it wasn't affecting the very early stages. So we were wondering then what could be affecting the early stages. So the mother has put in a lot of RNAs, a lot of proteins, and so we were wondering if those RNAs had to be translated. So we took a similar approach by injecting a translation inhibitor, in this case pyrimycin. And what I think you can appreciate is from the very beginning, it's not normal, right? You don't get the correct accumulation of actin or the correct patterning. You get actin accumulating in the center of a wound. So we could see that from the very outset, it looked like you needed translation. And this was a little bit of a surprise because these events are happening within minutes, right? And usually translation takes some amount of time. So it's a little bit of a surprise that translation is required. And we don't know yet what the genes are that are being translationally controlled, but now that we have the knowledge that translation is involved, we have the ability to go back and identify what those genes are. What we do have, though, are all these different genes that are transcriptionally up and down regulated in response to wounding at the later time point. So the ones that are down-regulated, up-regulated. And we wanted to know more about what they were doing. And so we took the top 15 and upregulated the top 15 downregulated genes, and we knocked them down and looked to see what happened in wound repair. And I'm just showing you a couple of them here. And what I hope you can appreciate just looking at an actin reporter is that they're not normal. In fact, all 30 that we tested have problems with wound repair, and they can range from overexpanding the wound, actin accumulating in the center, not being able to disassemble their actin ring at the end of wound repair, So it says to us that transcription is required for many of the different steps of wound repair, not particularly the initiation ones, but many of the other ones. And what we can do is take this collection of genes and look at them in many different ways. And so one way that we do that is to measure particular parameters of their phenotypes. And what I'm showing you here is just looking at contraction rate. So when the wound heals, we can measure the rate at which it contracts. For example, here, these are the 15 upregulated, the 15 downregulated. There's a control right here, and the pink bar that you're seeing is the 95% confidence level. And what you can tell are there are definitely ones that go slower, ones that can heal faster. And by measuring many different parameters of the wound repair, we can get sort of a combinatorial code of phenotypes. And that allows us to place them in the pathway, to figure out which ones are acting first or second, or the major types of events that they're affecting. And we've been able to do this with individual genes and to identify different pathways of genes. And one of the ones that was pretty surprising to us was actually insulin signaling. So we found two different genes, one at the top of the pathway, one at the bottom of the pathway in this. And insulin signaling itself is not new to wound repair. It's been implicated many times in tissue wound repair. But in single cell repair, it seems a little counterintuitive because you would have to then signal out of the cell and back into the cell. And that would take a lot longer than just maybe making a signal that goes within the cell. So we're really interested in understanding if insulin signaling was really involved, or whether maybe these proteins were moonlighting and having some role outside of their role in canonical insulin signaling. And so to start with that, we took a reporter of insulin signaling, which is the PIP3 gene that's used quite often to look at insulin signaling. And we looked at it after wounding. So here's a wound with actin, and you can see that PIP3 is very robustly recruited to a wound. And just to ensure that this is due to its role in insulin signaling and not, say, a moonlighting role, we knocked down the pathway by knocking down the protein upstream Chico. And what we find is now you don't recruit it to a wound, right? So this is responding to insulin signaling. One thing that we also noticed is that the actin is not okay in these mutants. And that gave us a little bit of a hint as to what this could be doing, and I'll come back to that. So we looked at many of the other points in this pathway that we had reagents for, and I'm showing you the chymographs here just with actin regulation. And what I hope you can appreciate is that they all affect wound repair, and their phenotypes are very, very similar, which is what you might expect if they're in the same pathway. And we can see that they're, for example, accumulating actin and having roles in delaying the wound repair process. One thing that we wanted to know is since the canonical pathway seemed to be working, what it was doing downstream, like what the effectors would be. And there are a couple of effectors that are known to affect actin dynamics, which is what we were seeing here, and what we were seeing in the Chico mutant. And so we looked at two of these. One of those is called GERDin. This is an AKT substrate, and it's known to bind actin filaments. Another is Chickadee, which is the Drosophila profilin homolog, and this binds to actin monomers and often works with formins to affect linear actin polymerization. And what I'm showing you here is just two examples of wounds and looking at the recruitment of GERDin and profilin. And you can see that GERDin is recruited, and so is profilin to the center of the wound and then sort of in a halo here. And as I showed you before, these are dependent on the insulin signaling pathway working. So if you knock down Chico, now you don't get accumulation anymore. So this led us to a model for how we thought the insulin signaling could be working in single cell wound repair. Oops, sorry, got ahead of myself. Both of those mutations also affect wound repair. And they do it in slightly different ways. So these phenotypes are affecting wound repair, but not the same. So for example, you can see in this case that the GERDin mutant is able to overexpand. It delays wound repair by shifting to the right. And the slope of this line and this line is very different. And so they're not healing at the same rate. So this did lead us to a model then for how this could be working. So here we have a cell. This is the perivitellin space, a space right outside the cell before the vitellin membrane, which is that impermeant membrane I was telling you about. When you breach the membrane and the cortex using a laser, calcium flows in, and this induces endocytosis. And endocytosis, one of the things that's going to be endocytosis is insulin-like peptides into this perivitellin space. And they will bind to the insulin receptor and then initiate the pathway that will give you activation of the downstream effectors. And it turns out that the distance from the wound that the insulin receptor is, you can get different downstream effectors to be activated. And this turns out to be very important because these different downstream activators that regulate actin do it in a different way. And right near the wound where you have the actin ring, you need a very dense organization of actin, but in the, say, the actin halo and further out from the wound, you need a less dense actin organization. So in order to get these different organizations and to be able to make them dynamic, you need to be able to quickly regulate these different downstream genes. So one reason for having this, you know, the whole point of releasing the peptide and then having to signal back in is to control the spatial dynamics. Okay, so what I've told you about our microarray screen is that we learned that transcription is not really required for the initiation of wound repair, but translation is. We identified 253 genes that are involved in wound repair, and these are allowing us to identify individual genes, families of genes, and pathways that we haven't really known about before that are affecting this process. And I showed you an example of one of those, which is insulin signaling, which is required for actin dynamics. And I'll just briefly tell you about the other one, the screen we did, which is a fly trap screen. And in this case, it's looking at GFP tag proteins that are recruited or depleted upon wounding. So in the fly community, there have been a number of transgenic flies that have been made that have GFP fused to proteins of interest. And we looked at 1,400 of these and found 141 lines where you see recruitment of the GFP signal, which is shown here, upon wounding. So this is just showing you actin for the wound. This is the GFP signal. And you can see ones that are just in the middle region where the membrane plug is, like here. Ones where they're in the plug, but they're depleted from the ring. For example, this one makes a ring, but it's actually inside the actin ring, or overlapping the actin ring, making an actin ring in a halo, or just the halo. And we get some other random patterns as well. So this gives us a number of different genes that are all involved in the wound repair process. And we can see not only the spatial patterns, but also temporal ones. So for example, here's one that is robustly recruited very quickly after wounding, but by the end of wound repair is gone. And this is one where it doesn't become really robust until the end of repair. And this allows us to tell when in the pathway they might act. So for example, this one that doesn't come up till later isn't a very good candidate for a start signal. But we do have some that come up as fast as, say, 3 seconds, and those are ones that would be. So where this is leading us is to a pathway that exists for all of the different models of cell wound repair, where we have the morphological events that are occurring. But now we have almost 400 new genes that are involved in the process that are going to let us match the molecular pathway and establish it to match this cellular one. And we'll be able to look at, for example, different events like membrane plug formation or the remodeling events that happen at the end, or different protein families like Rab GTPases or annexins. And so we're pretty excited about the future because we have a lot of things that we think we need to get working on. And I'll just end by acknowledging the wound healing team that have done all the work that I've shown you here. Our collaborator for bioinformatics, Jeff Delro, other lab members who do things that are a little bit different I couldn't tell you about today, and then our community resources and funding resources. And I'm happy to take any questions. Thanks.
Thank you, Susan, for a great talk. We have a few questions coming in for the Q&A panel. If attendees would like to ask questions, please submit them now. One of the first questions we have is, how functional are fly embryos how functional are fly embryos or the adult flies with the RNAi knockdown in the absence of a wound? Does perturbations of these pathways yield inviable embryos, implying a lot of background wear and tear that must always be fixed? Or are they fine, implying that these pathways are for a true crisis?
Well, the RNAi works really well in most cases. we can see really good knockdown. So in all the cases, we have to check how much knockdown we have, because in some cases we don't get great knockdown. In our particular system, we're knocking it down in the mother because we're looking at embryos that have just been laid by the mother. And so there can be cases where you knock down something by RNA and you don't get embryos. And that could be for reasons of stress. It could be for a lot of reasons. I don't think that we know. I'm sure there are examples of all of those. But they work pretty robustly in our hands for looking at wound repair. Because if you can get an embryo, the one good thing about wound repair is it's an inducible system. You induce a wound, you look at it right away. So you're not really subjected to all of the pleiotropic effects that you get when you're looking at a genetic process. So I think that answered many of the parts of that question.
We have one more. Can you titrate the response by transiently putting eGTA in the perivitreal space? Could Chico be stimulated in an insulin-independent manner?
We've not tried that. I mean, you can do those types of experiments. We've definitely injected inhibitors into the perivitreal space or into the embryo. For example, when we were looking at calcium, we could put BAPTA into the perivitreal space to look at what external calcium could flow in versus injecting into the embryo itself. So it's certainly possible to do all those, but in that particular case for the insulin signaling pathway, we haven't looked.
I think we have time for one more question. Does the size of the membrane lesion affect the cellular response?
It does and it doesn't. Up to a certain point, the response is scalar, so it's linear. When you reach a certain point, the dynamics change immediately, and that has to do with the stress response. So when you get too large, you induce a stress response, and when that happens, the dynamics change, and it would be the same as if you say burned your hand or burned half your body. In the case of your hand, you're just going to undergo wound repair. If you were in shock because half your body is burned, it would create a different pathway. So we try to keep ours below the stress response, but actually looking at all of these same things with the stress response is a very valid way to look at what happens as well, because both happen in nature.
Thank you. There are a few more questions in the question and answer panel. So if you wouldn't mind typing out the answers, we really would appreciate it. And thank you again for a really great talk.
Thanks.
I would like to introduce our next presenter, Dr. Derek Applewhite. Dr. Applewhite is an associate professor in the biology department at Reed College in Portland, Oregon. The central goal of his research is to understand the regulation of the cytoskeleton by addressing the following key questions. How does actin microtubule cross-linkers regulate the cytoskeleton? And how is actomycin contractility regulated during interphase? The Applewhite lab uses Drosophila flies embryo and Drosophila-derived cells as a model system to address these questions. Today, he will tell us more about the role of split discs in the regulation of non-muscle 2 contractility. Please welcome Derek.
Great, thank you. This is a beautiful picture of Reed College. Reed College is located in Portland, Oregon, just down the street from the Allen Institute. And so I'm very happy to share the work of my students, if I have control.
Yep, do one test click on the slide and you should be able to advance after that.
Do one, I'm sorry, one.
Try clicking on the slide.
Oh, okay.
And then you should be all set.
Oh, there we go. Okay, great. Great. So before I get too deep into the weeds, I want to thank the people who actually did the work. I'm just their mouthpiece. So Aidan Turan is a current postback in the lab. Most of the work I'm sharing with you today was his senior thesis. JJ is a assistant researcher. He's actually a student at WSU Vancouver. Amy just left the lab. She was a postback for 2 years, but now is at a graduate school program at University of Washington. And Sushruta is a current senior. He's actually a biophysics major. So I'll be sharing some of their work today. So the central star of my talk is non-muscle myosin 2. And some of you might be very familiar with non-muscle myosin 2, but some of you might not. But non-muscle myosin 2 is critical to any cellular process that requires contractility. So here we have a beautiful example of two cells undergoing cytokinesis. Ooh, that looked, let's see if that will. Okay, well, maybe it's a little weird on my screen. I hope it's not weird on yours. And here another process, we just got a great introduction about Drosophila development. This is a little later in Drosophila development, but during this process of morphogenesis, cells are undergoing apoconstriction. And then the apoconstriction is driving cells inside of the embryo to allow for tissue development. So again, that apoconstriction requires non-muscle myosin 2 contractility. And then here we have an example of a Drosophila cell undergoing migration. And if we were to watch the cell for a little bit longer, you would see that the cell would actually be migrating across the screen. But again, non-muscle myosin 2 is really critical for the contractility required for cells to be able to crawl. And at its very basic basis, non-muscle myosin 2 is regulated by a phosphorylation event. So the regulatory light chain, which is the cyan colored little glob, is phosphorylated, causing the molecule to open up into a oligomer competent conformation. And then because it's a poor motor by itself, it needs to team up with other non-muscle myosin 2 motors to form oligomers that then bind to the actin cytoskeleton and then walk along the actin filaments, causing the actin filaments to slide across one another. And in doing so, those actin filaments are connected to other things and it causes contractility to happen, as we just observed in those 3 examples in the slide previously. So I like to, this is an image of a Drosophila S2 cell by turf microscopy. And I like to imagine what's happening at the very edge of the cell is that formation of those giant oligomers. Now, because this is turf and I can't for sure certain that these are individual non-muscle myosin molecules oligomerizing at this resolution, I still like to think that that's what's happening here. We're seeing the opening or the activation of non-muscle myosin filaments, and they're oligomerizing into these larger structures. A lot of people give talks and they talk about this beautiful hypothesis they start with, and this is kind of not how this project started. This is kind of a look under the hood. So I kind of think power of scientific exploration, or AKA, that looks funny. So in the middle of my postdoc at UNC, I had a little lull in research in between papers and such. So I did a bioinformatic query about for proteins that had this calponin homology domain. So at the time I was working on another protein that has CH domain, and I just wanted to see what other proteins that were out there in the Drosophila genome that had them. And I found one protein that CG13366 that was uncharacterized that had CH domain. And I was interested in it because it was uncharacterized and not a lot was known about it. And it turns out briefly after I started to investigate this protein, I cloned it and transfected it in cells. A paper came out that was sort of described it a little bit initially. So it's known as split disc in flies, but it happens to have a very interesting story along with its human homologue. And again, this is another example of where you just have to go where the science takes you. So the human homologue is this protein called SPECC1L, which is kind of a complicated name. It's sperm antigen with coiled domains like 1. And here's a protein diagram of this of the protein. You can see at the very C terminus is that CH domain that initially piqued my interest. But the protein is has coiled domains interdigitated with linker domains. And it's putatively a cytoskeletal interacting protein. So CH domains, calponin homology domains, often bind actin, but also on occasion microtubules. So they've been known to interact with the cytoskeleton. And SPECC1L in particular has been associated with the spectrum of craniofacial disorders, including facial clefts, wide eyes, and incomplete closure of the esophagus. And these two mutations in particular, this Q415-P and G1083-S, have been associated with 2 different families that have these phenotypes. And while these phenotypes seem quite unconnected, in fact, they are united by cranial neural crest cell migration. And so again, initially interested in CH domain, and now here we're talking about cranial neural crest cell migration. So things, escalate really quickly. Okay, there we go. So cranial neural crest cells emerge from the hindbrain by delaminating from the epithelium, neuroepithelium, and they migrate long distances following distinct pattern, and they migrate through a variety of extracellular environments. So that's really important. They're also a really good example of collective cell migration. because they maintain contact as they migrate along these paths. And so again, cranial crest cell migration only happens in vertebrates. So this is not a Drosophila specific thing. But if they fail to migrate and get to the right place at the right time, they lead to a whole spectrum of morphological abnormalities in the face and the neck and the cardiovascular system. So mutations at SBCC1L cause this variety of diseases because, or pathologies, because this population of cells are not getting to the right place at the right time. And in development, timing is everything. So the human homologue of this protein binds to both, was putatively binds to both actin and microtubules. So in the top panel we have, this is taken from the first paper that described this protein. The top panel is a GFP tagged version of the protein, and we're seeing co-localization between GFP tagged SBCC1L and tubulin. And the bottom we're seeing immunostaining where they made an antibody against it and they co-stained with actin. So putatively this protein is cross-linking both actin and microtubules. But let's talk a bit more about the Drosophila homologue. So the amorphic alleles are not completely lethal, so some of the flies do escape, and they have a variety of phenotypes, most notably cleft proboscis and wing blisters. And what's interesting about this is because flies, again, don't have cranial crest cells, but they still have cleft proboscis, which mirrors the phenotype we see in humans often with people who have mutations of this disease. But what this collective, and so here's an example of, on the top we have a wild type fly, and then the bottom we have a mutant of split discs, and we see blisters in the wings, we see crumpled wings. The arrow in J is pointing to a cleft proboscis, which is kind of difficult to see, and then a crumpled wings. And all of these phenotypes suggest aberrant cell adhesion and migration. Oh, sorry, the words were cut off there. So despite the fact that flies don't have cranial crest cells, this protein's functions seem to be somewhat well conserved even in Drosophila. So what made me go, huh, that's funny? Well, like I said, I cloned this protein and I threw it into S2 cells and I expressed it, and it appears that instead of co-localizing well with actin microtubules, this protein appeared to mirror that of non-muscle myosin 2. And I further took a step further and looked at microtubules in conjunction with non-muscle myosin 2 by immunostaining. And in my hands, I don't see a distinct microtubule association of this protein. It looks like it's very much associated either with actin to a lesser degree, but mostly and most strongly with non-muscle myosin 2. And we can even look at the dynamics of the two proteins together. So here I'm expressing an mCherry tagged version of split discs, and then spaghetti squash, which is the regulatory light chain of non-muscle myosin 2. This will be my marker for non-muscle myosin 2 throughout the rest of the talk. And you can see the dynamics of them are very similar. And in fact, if we disrupt non-muscle myosin 2 by, in this case, we depleted the essential light chain, we see sort of a blowing up of that higher ordered oligomer structure of non-muscle myosin 2 in these cells. And you can see it's kind of dispersed. We see the same dispersion of split discs, which suggests that if wherever non-muscle myosin 2 goes, this protein split disc goes. So at this point, I was pretty convinced that split discs does co-localize with non-muscle myosin 2. And so we next set out to figure out exactly how. And so we took a classic structure function approach. And we used a very common technique called Mander's overlap coefficient. This expresses the degree of which two structures spatially overlap and takes into account the total amount of abundance of 4 4s that overlap and results in two values that range from 0 to 1. So in this example I'm giving you here, we have two discrete proteins, a cyan protein and a yellow protein, and they're happen to be in the same place at the same time. So therefore we have two high Mander's coefficients of this M1 and M2 value. They're both high, which suggests that there's some co-localization. Where on the right, we have an example of a cyan protein that is very discretely localized and then a yellow protein that is everywhere. And so everywhere the cyan is, there is yellow, but not everywhere where their yellow is there cyan. So we'll have an asymmetric distribution of these Manders coefficients where one will be high and the other will be low. And again, this suggests that there is not overlap, a lot of colocalization between these proteins. And so I use this, my student Aiden used this assay to assess different domains of split discs and see how well they overlap with non-muscle myosin 2. And the first question we wanted to know is if non-muscle myosin 2 in split disc preferentially associates with open or closed non-muscle myosin 2. And again, I told you in the beginning, non-muscle myosin 2 has to be phosphorylated, and it opens up into a oligomer composite conformation. And so we co-express the two mutants of non-muscle myosin 2, AA and EE. This is a non-phosphorylated form of non-muscle myosin 2, and then a phosphomimetic form of non-muscle myosin 2 with immature split discs. And to our eyes, it looks like there is a pretty good colocalization, but we used our assay to quantify this. And what we see is that the overlap between non-muscle myosin 2 and split discs is unaffected by the phosphorylation state of non-muscle myosin 2. However, the overlap of split disc with non-muscle myosin 2 is actually affected. So it looks like a split disc has a slight preference for open or active non-muscle myosin 2 in this case. We then just started to chop up the different domains of split disc to see how it colocalizes. And here I'm showing you an image, a movie of split discs that has a deletion in the first and second coiled domain compared to non-muscle myosin 2. And you can see we can delete all the way up to the second coiled domain and still see robust colocalization. And again, we quantified, oh, sorry, that's out of order, but that's okay. However, then we chopped off that CH domain, which was that domain at the very end of the molecule, which first piqued my interest in this molecule. And now we do see an uncoupling of non-muscle myosin 2 and split disc. So the top panel is a wild type split discs with non-muscle myosin 2, and the bottom is that delta CH, that calponin Himalayan domain. And now we do see an uncoupling. So if we quantify this again, we can see that we can delete all the way up to that second so-called domain and not see a difference between the amount of overlap between these two proteins. But then if we do chop that C-terminal CH domain, now we see an uncoupling. So it looks like that CH domain is required for non-muscle myosin 2 co-localization. Naively enough, I was like, well, maybe we just express that CH domain and that would be enough to co-localize with non-muscle myosin 2. And that now we've kind of figured out the key to this molecule, but in fact, I was wrong, which is not surprising. If we just express the CH domain of split discs, it does not co-localize with non-muscle myosin 2. And of course, again, in retrospect, that makes a lot of sense. There's a lot of coiled domains. Even though we can cut off the first two coiled domain, we still left that third coiled domain. And so I think, you know, the hypothesis down the lab, which makes a lot of sense, is that this molecule actually acts as a dimer. And again, many CH domains are found in tandem, and those that are not in tandem really bind the cytoskeleton very weakly. So this does suggest that this molecule might be a dimer. But what's really important about this molecule isn't necessarily the deletions of these domains, but how this disease-causing variants localizes non-muscle myosin 2, since those again result in craniofacial pathologies. And so we made some analogous mutations in the fly version of SB1L, and that's Q26P and G915S. They're equivalent to those two families that have their pathologies. And we expressed those with non-muscle myosin 2. We saw, again, what appeared to be robust co-localization. And then again, we quantified. And what we see that there is slight differences in the ability of non-muscle myosin 2 to overlap with split discs. There looks like to be a slight increase with the Q266P mutation, and then a slight decrease with the G915 mutation. And then we can see that split disc overlap with non-muscle myosin 2 is slightly decreased when we look at the other inverse Manders coefficient. But what's interesting is that non-muscle myosin 2 has a slight preference for that mutation, and this will come into play a little bit later, so keep that in mind. It might be that this Q266P mutation is acting as a dominant negative. Okay, so far what I've told you is split disc may have a slight preference for the open conformation of non-muscle myosin 2, and we are following this up with pharmacological manipulations because these mutations are complete open and closed conformation of non-muscle myosin 2. So using the drugs will be a good complement to those experiments. Deletion of the CH domain appears to uncouple split disc from non-muscle myosin 2. And that G915S mutation appears to decrease the amount of co-localization between split discs, while the Q266P mutation may enhance co-localization. And the expression of CH domain alone does not co-localize with non-muscle myosin 2, which suggests that split disc is probably functioning as a dimer. But what's really important about this protein is its role in cell migration. And so co-localization is great, but really we want to understand this role in cell migration. And so again, like cranial neural crest cells must migrate to form these structures, and the pathology is caused by point mutants rather than the deletions. What is the role of non-muscle myosin 2 in cell migration? And then on top of that, what is the role of split discs in this regulation? And so non-muscle myosin 2 plays a key role in the formation of focal adhesions. And focal adhesions are these structures which cells use to communicate and to bind to the extracellular environment. And for cranial neural crest cells, this is particularly important because they have to migrate through a whole variety of extracellular matrix environments. There's over probably 120 different proteins that are in focal adhesions. They form these very hierarchical structures, but non-muscle myosin 2 is also really critical for their formation and maturation. So our hypothesis was, if delay. Oh, here we go. Sorry. There we go. Our hypothesis is if split disc plays a role in non-muscle myosin 2 regulation, it's possible that focal adhesions are also affected. Okay. So we first looked at focal adhesion assembly, and so we depleted split discs and we compared to control RNAi treated cells. And again, this is a D25 cell. This is a Drosophila epithelial cell that we've expressed intracellular vinculin, which is a marker for focal adhesions. And we can measure the appearance of these focal adhesions, these markers. And we see no difference in the rate of focal adhesion assembly. So split disc doesn't affect focal adhesion assembly. But if we look at disassembly, now we see a difference. So RNAi depletion of split disc actually increases the rate of focal adhesion disassembly. So interesting, okay, they are disappearing more quickly. But what's really critical about this protein isn't necessarily RNA depletion or loss of split discs, it's gonna, or SPDC1L. What it is these point mutants. So then we next depleted the endogenous split discs, and we're able to add back these point mutants. And what we found was that, again, focal adhesion assembly was not affected by RNA depletion of split discs or by expression of these point mutants, but the disassembly rates of focal adhesion was greatly affected. So if we look at the Q26P mutation, we see that there was almost a 1.75-fold increase in the rate of focal adhesion disassembly rates when we express this mutant. And you can compare that to wild type or the G915S, which had no effect on focal adhesion disassembly rates. So now we're also able to kind of pick apart how these two different mutations are affecting the protein. Again, like as I alluded before, non-muscle myosin 2 appears to more readily co-localize with the Q266P mutation. And now we see that that mutation is specifically causing an increase in the rate of focal adhesion disassembly. And this is important because, again, as I said before, timing is everything in development. So if those cranial neural crest cells aren't getting to the right place at the right time, and that is dependent on their ability to migrate and their association with the extracellular matrix, that could lead to the defects that we see with people with this pathology. Yeah, again, so that 1.75-fold increase into focal adhesion disassembly rates because of that expression of that Q266P mutation. Sorry, a little bit of delay, which they warned me about. Okay, but now it's not responding. Okay, there we go, cool. So we wanted another way to be able to test the contractility by non-muscle myosin 2. And so what we used was a technique known as traction force microscopy. And this is where my collaboration with students in the physics department at Reed College have come into play. Traction force microscopy allows you to measure the forces of cell migration. So we're looking at a field of beads that are embedded in a polyacrylamide gel. And then on top of this, what you can't see, was a cell. And so as that cell migrates, it's pulling on the substrate and is moving the beads. And so the bead movement can be used to back calculate forces. And so my student, Sushruta, who worked with me this summer, was able to get this technique to work in the lab and started to measure forces by cells who have been depleted of split discs. And so here's kind of some of the data he got back. On the left, we have a vector map of the forces these cells are producing. And on the right, you're seeing, again, a heat map of those stresses. And what he preliminarily found is that if you deplete split discs, we actually see an increase in traction stresses. And so, so the next question that we're gearing up to answer, and this is a little bit more complicated experiment, is to, again, assess what these disease-causing alleles do in terms of the stresses forces the cells make when they migrate. So in conclusion, splitless co-localized with non-muscle myosin 2, and that CH-domain is needed for this co-localization. Depletion of split discs increases the rate of focal adhesion disassembly, and specifically that expression of that Q266P variant further increases focal adhesion disassembly. And then depletion also may lead to increases in traction stress. And in data I couldn't show you today due to time, we also know that depleting split discs in our Drosophila cells also decreases the rate of regular random cell migration. So all this is sort of tying in together about this connection between ECM, traction stresses, non-muscle myosin 2 regulation, and split discs. And just want to again give credit to where credit's due. This is my lab group before we had to disperse because of the pandemic. And I want to thank my funding agencies, National NIH, NSF, and of course Reed College. And again, all the people that worked on this project. Also, Peter Bar-Gillespie at OHSU, who also has helped with mass spec. Again, some experiments that I didn't have time to share with you today. And I will gladly take your questions.
Thank you, Derek, for a great talk. We have time for one question. Does expression of mutant SPDS forms affect the localization pattern of myosin 2 itself?
That is a great question. We have looked very hard at both depletion and overexpression of these mutants. And we have not seen a huge difference in the patterns, but I also will have to say that in S2 cells and even in the Drosophila cells, there isn't like a really consistent pattern. So I think if there are differences, it's quite subtle, but it's going to be hard to sort of figure that out because particularly in those migrating cells, myosin is just kind of lighting up like fireworks everywhere. So to find some sort of pattern that is kind of difficult. But that's a great question.
Great, thank you. We have to move on in the sake of time, but there are a few more questions in the Q&A panel. If you wouldn't mind answering them, Derek, we really would appreciate it. Our next talk is going to be a team talk by researchers at the Allen Institute for Cell Science. One of the missions of the Allen Institute for Cell Science is to understand the organization of a normal cell. The Allen Institute for Cell Science team members today will discuss how they used our single cell image data set to create a data-driven quantitative rules for the localization, amounts, and variation of 25 key cellular structures in stem cells. This allowed our team to create a baseline for normal cell organization. I would like to welcome our first speaker of the team, Callista Yang. Welcome, Callista.
Thank you, Irina. Hello, everyone. Today, Matthias, Ravik, and I are excited to present to you our recent work at the Institute with a study of robust integrated intracellular organization of the human iPS cell. In this initial study, we wanted to take a closer look at the organization of organelles and subcellular structures and to quantitatively and computationally characterize where, how much, and how variable they are in a cell. While the three of us are presenting this work today, we want to emphasize that this is a project of the collective work of everyone at the Allen Institute for Cell Science and 5 years of hard work dating back from cell line creation, data collection to today, the computational analysis and data release. Okay, so as you've heard multiple times, our goal as a team is to be able to look at a cell to know what it has done, what it will do, and what it's doing. There are a lot of different ways and different orders to look to characterize a cell, ranging over 4 magnitudes of scaling from gene expressions to global cell phenotypes. To decouple the complexity of cell behavior, we took the approach to the study of 25 intracellular structures that make up most of the main compartments and key functional units of a cell, and see whether we can build a framework for cell behavior with interpretable and testable set of quantitative constraints with 3D geometry of organelles. Now we know that cells are dynamic and a lot of times look very different from its neighbors, even the cell right next to them. When you look at the results of a lot of cells that look different, you begin to ask questions such as, what is the normal baseline variation that cells can take? In this work, we want to present to you the approaches we took to represent this population of normal, putatively identical cells with a full range of normal variation and embrace the variation that any cell can take. We use the Allen cell collection, a series of isogenic clonal HIPSC lines, in which each cell line expressed a single fluorescent tech protein for a specific organelle or subcellular structure through endogenous CRISPR-Cas9 gene editing. These cells underwent extensive quality control to ensure that other than GFP tag, there's no significant variation in gene expression or behavior. If you'd like to learn more about how you can use these cell lines for your research, we invite you to join the Allen Cell Collection Q&A later at 12 to speak with our stem cell experts. Now, among the whole collection, we selected 25 cell lines shown to you on the right that contain the key organelles of a cell, roughly ordered from the inside of the cell to the outside. We hope to image enough cells in 3D and in high resolution to measure the cell-to-cell variation in cell organization. We automated the culture of HIPSCs on robotic platforms and standardized image acquisition on a spinning disk confocal. These HIPSCs grow in colonies, and therefore for each cell we imaged, we also measured and quantified colony features, such as the size and where the colony the image came from, for example, the edge, the ridge, or the center. Overall, with this pipeline, we collected more than 18,000 images for this high-quality data set. For each of these images, which we call FOV abbreviated for field of views, we have 4 different channels: a bright field channel, a cell membrane dye channel to show the plasma membrane of the cell, a DNA dye channel to show the position of the nucleus, and the GFP channel to show the structure of the particularly labeled cell line. For example, here, Golgi is labeled. We then segment the fluorescent channel images using the Allen cell and structure segmenter to obtain information of boundary location, size, and positioning of the cell, nucleus, and the structure. If you'd like to hear more about this computational tool, feel free to drop by at the Allen cell and structure segmenter Q&A. With this segmentation, we're able to separate individual cells in the image and process them computationally and individually. For each cell, we quantify the characteristics such as the height, volume, shape of the cell, and DNA. We are also interested in quantifying the distribution of GFP tag structure inside of the cell. Now, with this data set, we have 25 cell lines, 18,000 field of views, and we have more than 200,000 images of individual cells in high resolution and 3D, together with the engineered features that we released for public use in this data set. Now, all these 200,000 cells look very different from one another, yet they're all normal. The next challenge we want to solve is describing this normality. Is there one average cell that we can use to compare with other cells in other conditions? Can we describe this population variation of HIPSCs in a statistical and quantifiable manner? Next, Matthias will guide us through this next stage of the analysis.
Thank you, Calista. To make sense of this huge data set and answer the questions Calista just mentioned, we created a shape space for cell and nuclear shape that allows us to group together cells with similar shape. Okay, so the first step is to make sure that all cells are oriented in the same way. And we do that by aligning the cells in the X and Y plane and applying the same alignment to the nucleus. Next, both cell and nuclear shape are described in terms of spherical harmonics coefficients. And the more coefficients we used in the spherical harmonics expansion, the better we can represent the shapes. And we can quantify how good the representation is by inspecting the distance between the reconstruction and the original shape as a function of the number of coefficients we used. For the rest of this talk, we used about 250 coefficients for both cell and nucleus, which is enough to provide 1 voxel precision in terms of reconstruction. Okay, so now we have both cell and nuclear shape represented in terms of spherical harmonics coefficients, and now we can use those coefficients to reconstruct the cell and nuclear shape in the correct location. Now we have this framework we can apply to all the cells in our data set. And we can use principal components to reduce the dimensionality of this data down to 8 dimensions, which is enough to explain 70% of the total variance in the data. So we call this 8-dimensional space the shape space. To explore and interpret the shape space, we sample different regions of that space using map points that discretize the principal components in units of half standard deviation. We can set map point values and use the inverse PCA to obtain the corresponding spherical harmonic coefficients, and from the coefficients reconstruct the shape. And because for this particular case, we set all map point values to 0, meaning that we are at the very origin of the shape space, the shape that we get here on the right side corresponds to the mean cell shape of our entire data set. And just keep in mind that this data set is composed by over 200,000 cells. Okay, and what I show here is the 3D view of that shape and 3 two-dimensional projections of the same shape. Now we can visit different areas of the shape space by sliding the map points to different values. For instance, here, moving the 3rd principal component to 1.5 standard deviation, and use the same procedure to inspect what the shape in that particular area of the shape space look like. Now we can apply this systematically across all principal components to determine what each principal component is measuring in terms of shape. And what we found is the first principal component is related to height, the second one is related to volume, and so on. And now that we have assigned names for these principal components, we are going to call them shape modes. Okay, so as shown here in this scatter plot, and I said before, the first shape mode is related to height. And height by itself is related to the packing, the cell packing in the colonies that we imaged. The second shape mode is related to volume, which reflects the cell cycle which all the cells go through. The cells in the epithelial colonies, they are constantly pushing on each other. And the remaining shape mode reflects the physical interactions between cells and their neighbors. So cells are constantly changing their shape. And next we asked what happens to the organelles and the intracellular structures inside the cell as they change shape. So we developed a technique to create a parametrized representation for the GFP intensity inside the cells. This is a schematic of how this procedure works, but I'm not going to go into details. All you need to know about this method is that it allows us to morph real cells into map point shapes. Okay, and you can know more details about this method by looking at our paper on bioRxiv. Okay, so now we can use this procedure to morph cells that are near each other in the shape space into the same shape. And now, because all the cells have the same shape, we can average them together to create average location maps like this one for mitochondria. We can do the same for different organelles, nuclear envelope, nucleoli, mitochondria, Golgi, and peroxisomes. And now we can visualize them in an integrated way on the same shape. Okay, so now we ask how variable is the location of intracellular structures in cells that share similar shapes, and how similar are the location of any two structures in different cells. And now Ritvik is going to tell you how we use stereotypy and concordance metrics to address these two questions.
Thank you, Matthias. So, oops. Let me see if I can get control. There we go. So now that we have all of these different organelles that have been morphed into the respective map points of the shape space, which are interpretable, now we can begin to ask how variable is the location of intracellular structures in these map points. And more specifically, we can ask how the changes in cell shape affect these locations. So to do this, we first establish a baseline by looking at the mean cell in the shape space. And we set up pairs of morph cell images per structure. And we compute the 3D voxel-wise Pearson correlation score across these pairs. We can then average out all of these correlations to get an estimate of the stereotypy for that structure. So in this case, we get the stereotypy for the nuclear envelope, but we can do this for other structures like the mitochondria, for example. So this analysis gives us a distribution of Pearson correlations across 25 different structures where the average Pearson correlation or the stereotypy represents how variable is the location of that structure. So from this box plot, you can see that the most stereotyped structures, so the least variable, include the nuclear envelope and the plasma membrane. And these are effectively positive controls because they were used as fixed points in the morphing procedure. This is followed by the two nucleolar compartments and then the two ER markers. At the other end of the scale, the least stereotyped structures are the most variable. These include the more sparse and punctate structures, for example, the centrioles and the desmosomes. And this is followed by the endosomes and the peroxisomes. So this gives us an idea of the relationships between these structures and the mean cell. And now we can look at how cell shape affects this distribution. But when we do this, what we find is that the stereotypy is completely robust across the shape space. So what you see here is a heat map of the stereotypy value across these 25 different structures and across all of the 8 shape modes, where the center bin is the mean cell. And so the stereotypy is only dependent on the structure itself and not on any of the map points of the shape space. And so this was a very surprising result. The next question we could ask is how do two structures co-localize? So again, we establish a baseline by looking at the mean cell in the shape space. And this time we're looking at the average morph representations per structure. And we compute the 3D voxel-wise Pearson correlation score between these structure pairs. And this gives us a heat map of these correlations. We can then take this heat map and perform hierarchical clustering to determine the clusters that these structures belong to. And what we find is this purely data-driven hierarchical clustering that's based on the structure images alone can recapitulate known cellular compartments. So for example, there's a nuclear compartment where nuclear structures are positively correlated with each other and negatively correlated with cytoplasmic structures. The nuclear envelope and the nuclear pores are strongly correlated with each other and also show some overlap with both the cytoplasmic and nuclear structures. And then there are the two big cellular compartments for the bulk cytoplasm and the cell periphery, where there are many known concordances that we can see, and some more interesting ones, like the fact that the Golgi and lysosomes are strongly concordant, which is evident in the literature, which suggests that the Golgi supports lysosome localization. So now that we have this baseline understanding of the relationships between the structures, we can again look at how the concordance would vary across the map points of the shape space. And much like the stereotypy, what we see is that the concordance is also very robust across the shape space, and the concordance values themselves are only dependent on the structure pairs themselves. Now, this analysis was performed on our very large HIPSC single cell data set with over 200,000 interface cells. But what we found is that we can randomly choose 256 interface cells and get back 7 out of the 8 shape modes of variation for cell and nuclear shape. And this follows for all of the downstream analysis where, for example, we can show that the correct order of stereotypy can be determined from only 11 cell pairs per structure. And the correct order of concordance can be closely recapitulated with only 256 cells per structure. So what this means is that a significant downsampling can be performed on our data set. And all of these analyses that we described here can be performed on a number of cells that can be collected over the course of a few days in any lab. So to summarize our talk today, we've presented to you a fundamental benchmark of cell-to-cell variation in healthy, normal, undifferentiated HIPSCs. And the Allen Institute for Cell Science aims to democratize access to all of the major data sets, tools, and analyses that we've described. So for information about the cell lines, the plasmids, the analysis tools, and all of the image data, please refer to these resources available online. Now, in this study specifically, we showed that HIPSC is grown in culture as epithelial colonies can vary in shape through a very simple and interpretable shape space. We showed that the cell-to-cell variation in the positioning of organelles or the stereotypy depends on the structure, but it's very robust throughout the entire shape space. And this follows for the concordance as well, which we showed can be calculated in an unsupervised data-driven way to recapitulate known rules of cellular organization. So for more information about these analyses and for other insights into the impact of cell and nuclear size on the variation in cellular structure size, please refer to our bioRxiv paper available online. So with that, we'd like to thank all of the people that contributed to this work. There's many people. We'd like to thank you for listening and also thank Paul Allen for his vision, encouragement, and support.
Thank you, Callista, Matthias, and Ritwik for a great talk. We have a few questions for you. The first one is, have you, after having set up the cell state, looked at the more extreme outliers? Do those outliers ever form informative clusters?
No, actually, at least in terms of the shape space, the distribution of cell and nuclear shape do not form any cluster, which was very surprising for us at the beginning. But hopefully in the future, when we compare this benchmark with like some type of perturbation, we were able to see the appearance of clusters.
Another question that came is did you find that centriole positions, even for elongated cells, being highly variable surprising?
I think in general, the centrioles were the more sparse and punctate structures. And it was surprising to see that even in elongated cells, it was very variable. And so to really try to understand that, we tried to compute these correlations at different resolutions to see if the centrioles would be correlated differently at a downsample resolution. And it was consistently a very sparse correlation. So this was surprising.
And then we have one more question that is asking, how is your approach able to close the shape of the cell and the nucleus in C at the caps, given the poor quality of image data in that dimension, and particularly the challenge of reconstructing surfaces that are parallel to the XY plane?
Yeah, that's the magic of the Allen cell structure segmenter. We use very advanced deep learning based segmentations for both cell and nuclear shape. And thanks to that, we are able to get very nice bottom and top of the cells and nucleus. And please check out the Q&A later to know more about it.
Yeah, this is actually a perfect transition into our next series of talks, which will be 5-minute lightning talks by several team members of the Allen Institute for Cell Science. Each presenter will give an overview of a different tool or resource that can be found on our data portal, allencell.org. We won't be having a live Q&A question after those lightning talks. So if you have any questions about the tools, any technical questions, we encourage you to attend our live Q&A session right after this morning's program. If you're interested, there will be a link for registration in the chat window. Our first presenter who will introduce the Allen Cell Collection is Maggie Fuqua, and she's a member of the stem cell and gene editing team. Please welcome Maggie.
Hi, everyone, and thank you, Irina. Thank you all for being here today. My name is Maggie Fuqua, and I'm a research associate here at the Allen Institute for Cell Science. I'm going to tell you a little bit about our Allen Cell Collection that Rue touched on a little bit earlier. All of us here know that studying cell behavior is incredibly interesting, but also incredibly complex. And so our approach to studying these dynamic cellular processes, we chose to look at it from a dimension reduction approach, where instead of tagging every protein within the cell, instead we chose to tag the primary structures and organelles within the cell to connect the phenotype to the proteome and genome. And so over the last 6 years, we've been actively engaging with the community to identify which structures would be the most impactful. Then combining gene editing with live cell imaging, we can tag these endogenously tagged fluorescent structures through stages like the cell cycle, or since we're additionally using human-induced pluripotent stem cells, we can track them through this stem cell state as well as through differentiated cardiomyocytes or other cell types. Our approach to generating this collection needed to check a lot of requirements for us that Rue mentioned, but namely that they are able to be openly distributed so that we can share them with all of you. This line for us was the WTC11 human-induced pluripotent stem cell line. And then we employed CRISPR-Cas9 editing technologies, underwent extensive quality control, which I'll tell you about in a minute, to end up with these beautiful isogenic stem cell lines that highlight endogenous proteins within the cell, as you see on the right. So this cell biology QC that we take all of our lines through includes genomic screening to make sure that the tagging is precise, that the tagged protein is actually expressed properly and localizes to the correct structure, that they grow at a similar rate to the unedited lines. Since they're stem cells, we want to make sure that they still differentiate and are karyotypically stable, among other things. To date, we have 47 cell lines that are available through Coriell. And I'm showing some here that highlight key structures and organelles. We have some that highlight stress and epithelial characteristics, as well as a number of lines that allow us to take a deeper dive into the nucleus, including 2 that were available as of yesterday, RNA polymerase 2 and the polycomb repressive complex EZH2. We also have some lines that contain multiple edits, including a couple of lines that highlight different components of the nucleolus. And some of our most recently released lines are CRISPR-I tool lines, including dead Cas9 for knockdown experiments. We also have developed a 2-step strategy to tag transcriptionally inactive genes, which enabled us to create these cardiomyocyte-specific lines that you see here. And even though for bandwidth purposes, most of the images I've shown you have been stills, these are all live cells. And this endogenous gene tagging allows us to uncover a lot of dynamic subtleties found in migration, in cell cycle, differentiation, you name it. Our mission here at the Allen Institute is to accelerate discovery in the biosciences. And our hope is that these tools that I've told you about and that you're going to learn about from my teammates will help provide more flexibility in your lab as you study your own cells. Being an open science institute, all of our data, methods, and cell lines are available in one place through allencell.org. And you can access images and quality control data that we generated through our cell line catalog, as well as links to obtain our cell lines through Coriell. And if you prefer to do this gene editing in your favorite cell type, you can access a link to obtain those plasmids through AddGene as well. We have video tutorials, protocols, and publications to make work with our lines and tools easier for you. And if you still have other questions, please contact us on our forum. You can talk directly to us, or you can also engage with other scientists in our forum community. We are really grateful to be a part of this cell biology community and really appreciate your time today. I'm going to hand it off now to John Chu, who will tell you a little bit about our cell structure segmenter.
Hello, everyone. I'm Jianxu Chen. I'm a scientist at the Allen Institute of Cell Science. Today, I'm going to give a brief update on the Allen cell and structure segmenter. Earlier today, our team has presented our recent work on iPS cell states, and it requires accurate 3D cell and nuclear segmentation of over 18,000 images from 25 different cell lines. And in each cell line, a specific cell structure segmentation algorithm is also required. The challenge for the cell and nuclear segmentation is not only the accuracy but also the robustness. For structure segmentation, here is an overview. Here is a snapshot of some of the structures that we have dealt with. Build a tool that can achieve reasonable accuracy on all of this is not an easy job. To tackle this challenge, we developed the segmenter, which is a Python-based toolkit combines classic algorithms and deep learning. The first part of segmenter is the classic image segmentation workflow, which is a 3-step workflow. In each step, there are only like restricted number of algorithms and parameters to tune. After building classic segmentation workflows for many structures, we came up with the idea of lookup table for people to use as a reference manual. So users can compare their own data with the images in lookup table and find the one that most similar to their own data. Here, by similar, I don't really mean the name of the structure. Instead, it's very important to compare the morphology. So as soon as you pick a workflow to start with, there will be a quick start, and you can do a quick start on Jupyter Notebook to adjust the parameters that most suitable for your data. With the embedded viewer and instructions in Jupyter Notebook, this will help you go through this process as easy as possible. Beyond Jupyter Notebook, more platforms are coming in next year. So after the classic segmentation workflow, if you need to improve the accuracy or the robustness, the iterative deep learning workflow can be used. Starting from the preliminary segmentation, you can go through the curation and use the curated result to train your deep learning model. Here, the curation can be sorting or merging. Sorting is just like pass and fail to find out a set of images that has been segmented correctly. Merging is just like you circle out some areas where you can apply another algorithm to create a merge ground truth to train your model. So this is very useful to deal with cells in different stages. When it comes to cell and nuclear segmentation, we can combine the iterative deep learning workflow with our new training assay approach to obtain more biologically accurate results. So the idea of the training assay approach is very simple. So when you are trying to create a segmentation target for DNA dye, and we can use the Lemon B1 channel to guide us on the how to determine the nuclear boundary. This is the training assay for DNA dye. Similar for cells. So even though the membrane dye may not always have very ideal signals, but we can use CACs to guide us to make such decision. So this is the training assay for cell membrane. And this results in like more than 200,000 cells, which has been released on Quilt. And as we have mentioned earlier this morning, the biological findings from this data set can be found in our recent bioRxiv paper. For the method itself, we have released the source code and all the trained models. Make sure you check out our segmenter paper on bioRxiv, which has just been updated. For more questions, please join us at 12:15 on the Q&A section. Next, I will hand off to Dan about the visualization.
Hi. My name is Dan Touloudis, and I'm a senior software engineer at the Allen Institute for Cell Science, and I'm going to introduce Agave 1.0, which is some software we have built for volume visualization. Oops. So version 1.0 of Agave is focused on pure volume rendering and display. All of the images that I'm going to show in this talk were rendered with Agave. It is a desktop application, runs on Windows, Mac, and Linux, all three major platforms. Our preferred open file format at the Allen Institute is OME TIFF, and so it was originally built around that. It can also load other generated TIFF files from software like ImageJ, and it also can support Zeiss CZI files. The visualization is done with a physically based rendering algorithm based on volumetric path tracing. And a byproduct of that is that we can have also physically realistic camera controls. And we can do things like controlling depth of field, blurring, and defocusing effects. And you can see a hint of that in the rightmost image here, where as the image recedes into the background, it's starting to blur because of defocusing. And you have full control over these effects. You can completely shut it off or control as much blurring as you want. So here's a picture of the user interface in a typical interactive session. This is a real-time interactive viewer. You have a pretty wide parameter space of controls to control the appearance of the volume data that you're dealing with. You can control lighting and shadow directions and colors. You can control, there are several controls for dealing with thresholding of your volume intensity data. And of course, the physically based camera controls and many other things. One of the big motivations for Agave was to provide a high quality alternative to typical Z-stack viewing, where you're looking at one plane of your data at a time and kind of scrolling through it up and down. We think that this provides another way of revealing spatial relationships in the data when you have lighting and shadows, and you can tumble the volume in 3D in real time. So we can also script Agave. We can animate essentially any of the visual parameters in it, and we've used it on very large files. So here's I'm showing an animation of a time lapse of some cell nuclei that was all rendered out in 3D with Agave. And we have another image here, which is basically a demonstration of camera rotation and other viewing parameters. The color changes during this animation also from that pink to that greenish color. Another interesting thing to note here is that in this data set, it may be hard to see, but there's a dividing cell here, and the direction, the sort of orientation of that cell is kind of diagonal through the volume. And that's something that you can easily see when you have this type of lighting and shading and you're able to spin the volume around. And it's something that may be a little bit harder to discern in a Z-stack, 2D planar projection type of setting. Next slide. Yeah, so Agave is available to download now at allencell.org, and we have a separate documentation page for it. And if you try this tool out and you have comments or suggestions or need help with it or anything, we have our forums. We have a special forum dedicated for that, and we would love to hear from you. And it is also open source software, so there's also a GitHub page, and if you prefer to get in touch with us that way, you can use GitHub as well. And with that, I can hand it over to Blair to talk about Simularium.
Hi, I'm Blair Lyons. I'm a software developer and visualization artist at the Allen Institute for Cell Science. And I'm going to talk about our Simularium project. We're building a platform for visualizing biological simulations in the context of whole cells. So today, the Simularium project is announcing our first public release, which is a browser-based visualization tool, the Simularium Viewer. And I'm going to start by giving you some background about the overall goals for the project and how this tool fits in, and then show you what you can do with the first release. So the long-term goal of a Simularium project is to provide a place where biologists can work together collaboratively as a community to build consensus models for cell biology. Our goal is to make it easy to aggregate and directly compare computational models and experimental data, to use methods at different scales or models that were simulated with different methods to inform and validate each other, to integrate diverse types of data from all available sources, and to make it easy to annotate and discuss parameter sets and model results as a community, with a focus on facilitating efficient and informative discussions between computational and wet lab biologists. We're working towards this goal by building software tools for the community to use, including computational biologists, wet lab biologists, and educators and students. This first tool that we're releasing allows computational biologists and their wet lab collaborators to view the results of 3D spatial simulations online. So on the site, you can explore some pre-computed example models that we have posted in 3D. So you can interactively rotate them. You can also play through and see the dynamics of the model over time, and zoom in and click on individual entities in the simulation to see their structures more clearly. You can also highlight entities by type and investigate plots in that same window. And this viewer uses rendering techniques from computer graphics to help separate objects in the foreground from the background, and to render the appropriate level of detail to support making meaning from the view. And this is an example of how you can load your own data into this tool. And this is just the first version of this viewer, and we're going to continue to improve the rendering features and performance. So you can start, like I said, by exploring a few example models that we've posted. The first model is actin and clathrin-mediated endocytosis by Matt Akamatsu. And this model uses Cytosim, a community cytoskeletal simulation engine, to model a branched actin network internalizing an endocytic pit against membrane tension. The second example is a SARS-CoV-2 dynamics in human lung epithelium. And this model is part of a rapidly developing community effort to model the novel coronavirus. And it uses PhysiCell, which is a community cell scale simulation engine. So we've also provided tools and tutorials for converting your own custom data into our format, and you can find them here on the webpage. These tools include a Python conversion package, Simularium.io, to easily write your data to our JSON format in Python. We've also documented this format so that you can generate it yourself in your favorite programming language. And we're also working with authors of some simulation engines to add the ability to directly output to Simularium files. So here's just a few screenshots of some example models that we've visualized in Simularium. The Allen Institute for Cell Science is investigating the use of these types of simulations and the various simulation engines that generated them for building an integrated mechanistic understanding of human cells. So this is all available now at simularium.allencell.org. And I want to note that this is freshly released software. So if you run into any bugs, please try reloading the page. And if you can, let us know what happened by going to help submit issue. Like that in the top corner. And we'd also love to hear any feature requests in the forum, which you can also get to here. And with that, I'd like to thank my colleagues for their awesome work on this project and Paul Allen.
Thank you, Maggie, Chanshu, Dan, and Blair for a great introduction to our tools and resources. Just a reminder that we're not going to have a Q&A session right now. If you would like to ask questions about the tools and resources, please register for the separate Q&A session that we will have after the completion of the talks today. You can find that link in the chat window. We will now take a short break and we'll resume with a second set of talks at 10:15 a.m. Pacific time.
Hi, David. You're going first, right?
Yes, I am.
Okay, just trying to make sure. I'm Brock. Nice to meet you. I think we met maybe last year.
Likewise. Oh,
All right.
Sorry, just getting the script ready.
We'll get started in about 3 minutes, Brock, and I believe
Okay.
Calista is moderating this afternoon. You'll join at the panel.
Sounds good. So Callista, you're going to do this one, or?
Yes, I'll be emceeing for this session.
Okay, and then I'm going to do, so I'll just shut up. I'll shut up and go away. That's what everybody wants. Nice talk, by the way. That was nice.
Alrighty. Okay. So, hello everybody. Welcome back from the break. My name is Callista Yen, and I'm a scientific data engineer on the microscopy team at the Allen Institute for Cell Science and a member of the Symposium Planning Committee. I'll be the MC for the remainder of the day. Our next two presenters are from the inaugural group of Allen Institute for Cell Science Junior Fellows. The Junior Fellows program will engage emerging scientific experts to provide advice to the Allen Institute for Cell Science, introduce them to the Institute's science and culture, and at the same time, offer the fellows new opportunities for scientific development and potential research collaborations with the Institute. Unfortunately, Johannes Schoenberg is unable to join us today, but we'll hear from the other two Junior Fellows. The first one is Dr. David Van Valen. He is an assistant professor at the Division of Biology and Bioengineering at the California Institute of Technology. His research group's long-term interest is to develop a quantitative understanding of how living systems process, store, and transfer information, and to unravel how this information processing is perturbed in human disease states. His group leverages and pioneers the latest advances in imaging, genomics, and machine learning to produce quantitative measurements with single-cell resolution, as well as predictive models for living systems. Please join me in welcoming David.
Thank you for the kind introduction. So today I'll be talking about my group's work at the interface of biological imaging and artificial intelligence. I able to, oh, sorry, okay. So this interface between biological imaging and artificial intelligence has never been brighter. The number of things that we can measure about living systems with imaging has never been greater. Signaling dynamics, gene expression, protein concentrations, all of these are now measurable with imaging. At the same time, new deep learning, also artificial intelligence methods, have transformed the field of computer vision. As these methods make their way into the life sciences, they're fundamentally changing our relationship with imaging data by letting us extract quantitative information from these images with an ease that was previously impossible. A collection of tasks that can now be done are shown on the slide, which is meant to represent the field's progress, not just that of my group. These applications include cell segmentation, which is the focus of this talk, cell tracking, analyzing single molecule images, restoring noisy images, and interpreting label-free images. While there's a lot of excitement about these methods, AI hasn't had quite the same impact in biology as it's had in other areas of society. The reason for this is because we're currently in the Tycho Brahe era of AI-powered cell biology. What I mean by that is the following. Johannes Kepler was a famed astronomer. We all learn about him and his laws of motion during freshman physics. You know, the orbits of planets are ellipses, equal areas are swept out in equal time, and whatnot. These kind of general rules about how the world operates are the sort of scientific holy grail that inspires people, myself included, to go into science. What's less appreciated are the contributions of Kepler's mentor, Tycho Brahe. Tycho Brahe was also an astronomer, and he kept meticulous notes on the identities, locations, and movements of the planets and stars, compiled over decades. And it's exactly this data-centered effort, collection, curation, annotation, that enables the generation of knowledge and insight. And it's exactly in this, this phase that artificial intelligence and cell biology are in now. The challenges ahead of us as a field are substantial, but I say rather than avoid these challenges, we should embrace them. In my mind, there's really no alternative. The path to an AI-powered future for cell biology goes directly through these big data challenges, not around them. For the past 3 years, my lab has been tackling these issues, and we have a sense of what the data and the associated annotations of the future will look like. So the data itself needs to contain information about the parts list variation, number of genes and whatnot of living systems, as well as the spatial temporal variation that's just present in living matter. The annotations, so how these imaging data are labeled, they need to be performed at pixel level resolutions. You need dense pixel level labeling, and you also need to have single cell resolution as well. And a prototype, what I'm talking about, is shown in a video on the right. So here we have a nice confocal image of a cell undergoing mitosis, and that's paired with a annotation of mitosis, which is actually a diagram produced by the famous cell biologist Walter Fleming in 1882. And I think it's a little poetic that the future of this space actually starts sort of at the beginning of cell biology. Okay, so with that said, I'd like to share one story from my lab that we've recently finished. So this surrounds being able to do single cell analysis and multiplexed images of tissues. So new multiplexed imaging methods make it possible to image the location and abundance of dozens of different proteins simultaneously in tissues. These methods involve either imaging with mass cytometry or performing iterative rounds of staining and imaging with light. And the data they generate are opening new windows into the biology of both healthy and diseased tissues. So here we see, a panel of images from a set of patients that visualizes the difference in expression for a wide variety of markers. Now, these data are challenging to analyze, and single cell analysis is a common challenge for all of the imaging data that's coming out of these platforms. If you know which parts of an image correspond to individual cells, so saying these collection of pixels belong to cell number 1, these collections belong to cell number 2, cell number 3, and whatnot. If you have that information, then you can do analyses like extracting, and you can extract information like cell types or even spatial patterns. And so, here is sort of an example of what can be done with these sort of data. So this is an image of a patient's tumor, triple negative breast cancer, and all the different cell populations, tumor cells, different immune cell populations are identified in different colors. Now, most analyses done to date on this sort of data, even ones that I've been part of, use cell nuclei as a proxy for the whole cell. And this is inadequate for a variety of reasons. Most proteins aren't localized to the nucleus. And so if you're using the nucleus as a proxy, your quantification of those proteins is already biased. And moreover, if you only have nuclear segmentation, it's impossible to perform subcellular localization analyses. And it's also impossible to measure cell morphology. And really what this space needs is the ability to do whole cell segmentation. And so we set out to solve that challenge with deep learning. Our starting point was to collect data from different imaging platforms and tissues across the community. And this slide just shows a representation of what the data looks like. And looking at it shows both the opportunity and the challenges. So when we're compiling this data set, we made sure that each image had both a nuclear image and also a membrane and cytoplasmic image to ensure that the information necessary to perform whole cell segmentation was actually present. We also see the challenges. Cell morphology varies, quite drastically, both across and within tissues. Signal to noise and autofluorescence characteristics vary across the different imaging platforms. And then there's just the reality of being in tissues. Cells can be tightly packed, there's ambiguity, and you'll even notice that some cells don't even contain nuclei because the cell nuclei were missed by the imaging plane. To annotate this data, we developed a human-in-the-loop approach to data annotation. So here, experts, crowd workers, and artificial intelligence sort of work together to develop these data sets. Experts create enough data for a preliminary model and then perform quality control. Crowd workers correct model errors, and as the amount of quality data grows, models can be retrained, and these retrained models become more accurate, making the crowd workers' task easier. With enough data, you can train a accurate final model and then deploy it to be used by the community. And so we've finished the annotation, and the resulting data set we're calling TissueNet, the name inspired by the original ImageNet, which sparked the deep learning / artificial intelligence revolution. It's the largest collection of its kind. This data set is composed of images from 6 different imaging platforms: Codex, CISIF, Spectra, MIBI, multiplex immunofluorescence, and imaging mass cytometry, as well as 6 different tissue types. So immune tissues, GI tissues, pancreas, breast tissues, lung, and skin. And we have both normal and disease tissues in this data set as well. We've across all of these images, we've annotated approximately 1.3 million whole cells and a comparable number of nuclei. And we've done a survey of the literature, and we just to give a sense of the scale of this effort, this data set has more single cell annotations in it than what the entire field has done to date. With this data set, we can train deep learning models to perform nuclear and whole cell segmentation. We're currently calling this model multiplex deep cell, but this might change thanks to some of our more imaginative lab members. Multiplex deep cell works by predicting the cell contours and the cell centers. And once you have these 2 pieces of information, we can quickly determine which parts of the image belong to each cell. Okay, so in this case, an image is worth 1,000 words. So here, I'd just like to show you some of our results. Here we see an image of a pancreatic cancer specimen given to us by Travis Holman and Memorial Sloan Kettering. And when we run this through multiplex deep cell, these are the results. So the white outlines are our predictions of where the whole cells are in this image. So that was one sample. And so here's a collection of images from different tissue types, different disease and different imaging platforms and different disease states. And here are the predictions produced by multiplex deep cell. Each cell is colored by how much the cell area differs from the ground truth, white being no difference, blue and red being large differences. And so what we see is that our model is accurate across tissue types, imaging platforms, and disease states. And so I want to be clear, this is a single model performing all these segmentations. So we've also spent a considerable amount of time benchmarking model performance using a variety of metrics. And what we've observed is that multiplex deep cell is significantly more accurate on tissue data than existing methods. And it's also nearly an order of magnitude faster than the next most accurate approach. And so here on the left, we have a plot of accuracy given by F1 score and on the y-axis, and then the inference speed on the x-axis. Multiplex deep cell sort of gives you the best of both worlds. And then we also have plots of different accuracy measures. So the recall, the precision, and then also the Jacquard index, which sort of measures the actual quality of the cell masks produced. Okay, we've also compared multiplex deep cell to human performance. So we've observed that the difference between multiplex deep cell and human annotators is actually indistinguishable from the difference between different human annotators when evaluated on the same images. We've also done a comparison where we've asked pathologists to perform a blinded evaluation of multiplex deep cell's outputs compared to expert human annotators. And what we found is that pathologists were actually unable to display a preference between the two choices. So here we have a score of 1 being the human annotations are better, 5 being the AI annotations are better. And so what these two pieces of information sort of lead us to conclude is that multiplex deep cell is performing whole cell segmentation with human level accuracy. So we're just now digging into what we can do with such a model, and we feel that these results, these capabilities are going to be transformative for both tissue biology and digital pathology. So we're able to do protein localization analysis now. So here we see, there are different markers. So Ki67, nuclear, E-cadherin, present in the membrane. And so what we see is that, for a variety of different nuclear and non-nuclear markers, a multiplex deep cell is able to accurately identify that the nuclear markers are in the nucleus and the non-nuclear markers are not in the nucleus. Excitingly, we're also able to make cell morphology observable within tissues. So the study of cell shape has a rich history in cell biology, and we're excited about what it can do in tissues. Here we see that we can accurately predict a morphology feature. So here, like the nuclear to cytoplasmic ratio. And what we're also finding is that there's just rich information captured by cell morphology. So here what we've done is we've taken a variety of different morphology features, cell area, skewness, nuclear eccentricity, the nuclear to cytoplasmic ratio, and then performed dimensionality reduction using the UMAP algorithm, and then colored the different cells by their cell type present in their data. And so what we see is that there is information about cell type that's being captured by morphology alone. And what's exciting about this is that this morphological information is not biased by imaging platform, and so this makes us think that we can use cell morphology as a way to harmonize disparate measurements across tissues. So everything I just showed is very exciting, but I think what's even more exciting is what our work holds for the future. Because we've actually built the infrastructure to support projects like this, the marginal cost for producing new data sets is surprisingly low. And so we just calculated the total number of crowdsourcing hours required to execute this project. And what we found is that the cost for the next tissue net would be around $20,000, which is similar to a medium-sized sequencing experiment. And this low cost is due to both our human-in-the-loop approach and then the software infrastructure to support the full life cycle of this sort of effort. So from data collection and curation to annotation to model development and deployment, the ability to do all of these things in-house really makes the data set construction process much more scalable. So of course, models aren't any good if you can't share them. And so to share work like this, we've built cloud native software for hosting deep learning enabled image analysis pipelines. This software can handle large volumes of data, and what it does is it scales the cluster size to meet demand. If you have a large amount of data flowing through the cluster, it requests more resources from the cloud provider. And if you have less data, it requests less. And the scalability, this dynamic scalability is what enables us to both serve large numbers of people while also keeping the cost manageable. And so we envision the software as being like a blast for cellular images. Because the cluster lives in the cloud, users can just submit images for analysis and then receive the results, much like you can do with BLAST and sequences. And we've actually built plugins for image analysis ecosystems like ImageJ and QPath, so users can do this. And so this deployment model actually, we believe, solves many of the pain points of using deep learning in practice with a reasonable price of network latency. And so a example of this is shown below. We recently hosted a workshop at HGMI's Image to Knowledge tutorial, and one of our colleagues at the La Jolla Institute's Imaging Center tried our software. And so what they did is they took their own images of tonsils, opened it in QPath, used our plugin in QPath, submitted it to our web server. They went through multiplex deep cell, and then they got the results back. And so here you can see sort of the membrane stain in red. The blue-green is nuclear stain, and then the yellow are the predictions for multiplex deep cell. So we're really excited about what this is going to allow folks to do. We've made all the software that our lab produces open source. So it's available at our lab's GitHub under a very permissive modified Apache license. And we also host a web portal, deepcell.org. There are models that we've made that we feel are useful. And deepcell.org is a very easy way for people to try them out on their own data and see whether they work well for them or not. And I would say that the software tools that we've developed sort of are meant to manage like the full life cycle of deep learning projects. So from creating and annotating training data to developing new models to then deploying it in inference. We have software tools for all of those. All right, so that concludes my talk. I need to thank my entire lab. There is no one on this list who didn't contribute to this work. And without them, none of this would have been even remotely possible. I'd also like to thank collaborators, Michelangelo, who's been a partner in crime in our efforts to bring single cell resolution to tissue biology, and his student Noah Greenwald, who's effectively been a graduate student in our lab for the last couple of years as we've been developing this method. And Liat Karen at the Weitzman as well. She's been a fantastic collaborator. And last, I'd like to thank our sponsors, namely the Kersey Foundation, the Reed Allen Foundation, both the NIH and the Paul Allen Foundation, who support this work through subawards, and then also various centers at Caltech, both for funding and support. And so with that, open to questions.
Thank you, David. We now have time for a couple of questions that have come in through the Q&A. The first question is...
Okay, I think I see the question. Do you think there are techniques on the horizon that might allow segmentation of single cells in systems where a nice membrane label isn't available or feasible? Yeah, so I think my view of deep learning and sort of the whole supervised machine learning is that these methods really work best when the underlying data have the information within them, or the underlying images have the information within them to allow the models to make predictions. And so what you would need in this case is another imaging modality that contained membrane type information without having to have a membrane stain. If you had a modality like that, then I'd say like there are methods that you could do to sort of train a network to be able to do this. In the absence of that, I'm less optimistic, and I'd say nuclear segmentation is probably what would be the most effective in those cases. David, does this approach work when focusing on dividing cells? At the end of the day, you need to make a decision about what's one cell and what's two cells, and what's challenging about annotating tissue data is you don't have the dynamic trajectories, and so oftentimes like that's confusing. I would say that we have like human level accuracy, and we've sort of benchmarked it like across like all the different tissue types and the different imaging platforms. and they're almost certainly like mitotic cells captured by our data set. We are working on deep learning methods for analyzing dynamic live cell imaging data, and so we have a preprint that we put out about a year ago where we showed that we can actually annotate entire movies, not just static frames. And we've been able to train deep learning models to do both the segmentation and then also the tracking and lineage construction. So I think we're in the midst of sort of refactoring that work and preparing it for resubmission. But that's definitely something that's well within our technological capabilities. And so have you considered using synthetic data for deep learning model training? We have. I think there's some cases where it works, and there's some cases where it's less than optimal. So an example of where it works well, I'd say where the images are simple enough where you can write out like a reasonable generative model for image formation. So a good example of this was a Nature Communications paper. I forget the name of the PI, but they're at University of Virginia. And what they did is, they want to do segmentation of bacteria and biofilms. Bacteria have much simpler shapes, they're rods. And so they wrote a simulator to simulate what phase microscopy images would look like if you have like a sort of like a biofilm. For cells and cell culture to cells and tissue, I think like tissues, I feel like that's just not a viable approach. The heterogeneity is too great. We have tried some approaches like generative adversarial networks to try to use that as a way to simulate data. But I'd say like there are in our hands, like the success has been mixed. And given how, I don't want to say like how easy it is, but given like our ability to just get data annotated, it's more straightforward for us to just add data to the pile to be curated and annotated. I'd say single molecule fish data. That's another case where almost every deep learning method I've seen trying to do spot detection in single molecule data relies on doing simulations. What we found is that once you start moving into tissues that have higher autofluorescence, those simulation methods don't work as well as we would like, and more annotation centered methods actually tend to work a little bit better. So we have thought about it. It's a nice approach, but as I said at the beginning of my talk, I don't think there's any way around the need to do large amounts of annotation and curation.
Awesome. Thank you so much for answering the questions, David, and sharing with us your tools. There might be more questions coming through the chat, so please pay attention to that.
I will.
Thank you. In the interest of time, we'll move on to the next speaker. Our next speaker is our junior fellow presenter, Dr. Martin Kapman. Dr. Kapman is an associate professor in the UCSF Department of Biochemistry and Biophysics and the Institute for Neurodegenerative Diseases, and an investigator at the Chan Zuckerberg BioHub. The goal of Dr. Kapman's research is to elucidate cellular mechanisms of brain disease and to develop new therapeutic strategies. He co-developed the CRISPR-I and CRISPR-A screening technologies, and his lab has pioneered CRISPR-based functional genomics in cell types derived from induced pluripotent stem cells. Welcome, Martin.
Thanks so much for having me and for organizing a really exciting and diverse symposium. Yeah, let me see. Trying to advance the slides.
Click with your mouse to advance.
Okay. Okay, there we go. I think all of us are really excited about the diversity of human cell types and shape and function. And of course, one central question is how a single genome gives rise to so many diverse cell types. And generally that's thought to be because of different patterns of gene expression. And we now have ways to catalog those gene expression profiles in efforts such as the Human Cell Atlas. A question that I think has received less attention, but that I find just as fascinating, is the question whether not only gene expression determines cell type, but cell type actually determines the function of specific genes. So if you have a gene product Could it be that it actually fulfills very different roles in different cell types, such that, not only could it be essential in one cell type or non-essential in other cell types for various reasons, but that, for example, it could moonlight and have a different molecular role and cellular role in a different cell types. There are some exciting examples of that, but I don't think we have a systematic understanding of that. And a related point is that, there can be a very cell type specific impact of mutations that cause disease, even in broadly expressed proteins, for example, that affect very specific cell types and tissues. So while the human cell atlas currently is basically a human cell atlas of gene expression, it does not tell us or reveal these cell type specific functions. So I think really the next goal for cell biology should be also to generate a human cell atlas of gene function. And I'll tell you about two technologies and some results in our lab that I think could help us get there. First of all, we need to be able to perturb genes in a very specific way. The CRISPR technology needs no introduction, got recognized by the Nobel Prize this year. And in the early days of CRISPR development, a group of people at UCSF developed a number of technologies that involve catalytically dead Cas9 proteins, including the next speaker, Ron Vale. And when I was a postdoc in the Weizmann lab, I helped to develop genome-wide screening platforms that use this dead Cas9 tethered to transcriptional repressor or activator domains to tune up and down gene expression over many orders of magnitudes to look at how the expression level of a given gene affects cellular phenotypes, not only one at a time, but also in genome-wide screens. And while all of this was initially done in cancer cell lines, I was really excited when I started my own lab at UCSF to do this in non-cancerous, specifically differentiated cell types, and we decided to implement it in iPSC-derived cell types. And what I will tell you today is first a little bit of our technology platform that's published, but then also a vignette of unpublished data where we found a pathway that is a neuron-specific cell biology that I'm quite excited about. And both of those projects were led by a fantastic graduate student in the lab, Reilin Tian, who just left the lab a couple of weeks ago to set up his own lab in China. So we established a platform that allowed us to knock down or overexpress endogenous genes in human neurons, here looking at protein progranulin, for example. And we were also able to conduct large-scale pooled screens that helped us identify genes that are specifically essential for neuronal survival, but maybe less so other cell types. We were able to have high-dimensional readouts for perturbed gene functions, such as single-cell RNA sequencing and microscopy data. This is published work, so I'll briefly highlight just two examples that I found particularly interesting from this first methods paper. So as I mentioned, we can knock down genes and look at the transcriptomic consequences. And for each gene knockdown along the y-axis here, get a kind of rich fingerprint of how cells react and how gene expression changes along the y-axis. There are certain modules that are coherent, that make sense. I'm just going to highlight one single gene that I found interesting in this data set. UBA1 is the E1 ubiquitin activating enzyme in the E1, E2, E3 cascade of ubiquitination. It's ubiquitously expressed in the human body. It's essential in the cell types we looked at, and based on the literature, not surprisingly, giving the many roles of ubiquitination. But when we knock it down in either iPSCs or neurons derived from the very same iPSCs, the reaction of the cell was completely different. In neurons here on the bottom, we see upregulation of a lot of proteostasis factors, molecular chaperones, for example, in different compartments. And this is what I might have expected, given the importance of ubiquitin-mediated protein degradation to maintain protein homeostasis. In iPSCs, the picture is completely different. None of these genes are upregulated. Instead, we see a different signature. Metallothioneins go down, a signature that had been associated with oxidative stress. So this is just to highlight that knocking down the same housekeeping genes in two cell types can have vastly different consequences. Maybe it's limiting for different reasons in different cell types, or cells have evolved to respond very differently to the same stress. So again, highlighting the importance of studying gene function in the relevant cell type. Maybe actually more interesting to this audience is the results that we got by doing high content imaging when knocking down different genes and taking a time-lapse view of what it did to cells. This data was acquired together with Michael Ward at the NIH. On the left, you see how neurons over a couple of weeks normally differentiate with a non-targeting control guide RNA. On the right, this is one of the genes that was neuron essential, and you can see those neurons are not so happy. They die. They don't properly grow out their axons. And so you could say, so what? You know, this what happened to be in neuron essential genes. Neurons die and they're sick. And so what do we learn by that? Well, it turns out that we have kind of an Anna Karenina situations here where all the happy neurons are the same, but all the unhappy neurons are unhappy in their own way. And to find that out, Ray Lin actually did some image segmentation, pretty old school segmenting based on two-color imaging, the nuclei and cell bodies, and just quantifying a number of metrics, how many trunks come off of each cell body, what's the length of the neurites and the branching degree. And just based on those few metrics and looking at cell numbers over time, he looked at a data set of roughly 25 genes, targeted each by 2 guide RNAs. And these genes were selected because they were neuron essential in our large-scale neuron survival screen. They were not selected because they had any known effect on, say, neuronal development or morphology. But what we could see is that the 2 guide RNAs targeting the same gene, shown here in the same color, had over time quite consistent effects on neuronal morphology, and these effects were distinct compared to the non-targeting guide RNAs in black here. And so when we put that data together in a heat map where the different columns are the different metrics over time, and each row is one of the guide RNAs knocking down a specific gene, we get these fingerprints. We can cluster hierarchically. And one thing that really surprised us and that I found very exciting is that not for all, but for quite a large number of genes, knocking down the same gene with 2 different guide RNAs gave us very similar patterns just by looking at them by microscopy. So looking at these unhappy cells in a dish has enough gene-specific information to tell us, oh, those neurons have a problem with electron transport chain complex 5, those or ATP synthase. These neurons have a problem with cholesterol biogenesis. These have a problem with endolysosomal trafficking. So there was tons of information just looking at the cells. And it reminded me of this paper where people use machine learning, for example, to look at children's faces and were able to then predict the type of genetic disease that they had. So, you know, looking forward, I think it will be very exciting to look at several high-dimensional phenotypes, including morphology for neurons, obviously rich phenotypes such as neural activity, transcriptomics, and other cell-based readouts, and to try to integrate them, and also to not only be able to kind of predict maybe what of these features, you know, are defective in disease. And so, you know, we could take patient-derived cells, for example, where there's no genetically known cause for the disease and try to observe them to find out what cellular processes might be perturbed in these cells. And we can create a lookup table using the CRISPR-IA technology. But also later on, maybe to cross-map what are features in the transcriptome that would predict a very specific pattern of neural activity or a very specific aspect of morphology to then come to some mechanistic understanding how these cellular features are actually happening on the molecular level. So that was kind of the technology overview. We're very excited to compare this and extend this to many cell types. In unpublished work, we have this also in astrocytes and microglia, and to provide data commons that enable the community to search their favorite genes, their favorite cell types, and we created a first version of this called CRISPR Brain. We'd love for everybody to give feedback and suggestions for future features. And of course, if you're doing genetic screens in human cell types, we'd love you to connect and contribute data to this data commons, specifically to start building the types of cell types and also high-dimensional phenotypes that are represented. But now I want to end on an actual biological vignette. And we were asking a very specific questions about neurons. How do they deal with oxidative stress? Why was that something we were interested specifically in neurons? Well, reactive oxygen species are a normal byproduct of cellular metabolism. In particular, the #1 source is oxidative phosphorylation, and these reactive oxygen species can cause damage to macromolecules in the cell. And there's reason to believe that the brain in particular is very vulnerable to reactive oxygen species. While it only accounts for 2% of the body weight, it actually consumes 20% of the oxygen, and it is really chock full of polyunsaturated fatty acids that are very vulnerable to being oxidized. Within the brain, really, the neurons face a particular challenge. They're post-mitotic, they're among the longest living cell types in the body, and they are hooked on oxidative phosphorylation. In fact, when neuronal progenitor cells differentiate into neurons, there's a metabolic switch from glycolysis to oxidative phosphorylation. So we think neurons must have some specialized way to be able to deal with oxidative stress, and maybe in aging and disease, those mechanisms could break down, because we know that in an old brain and in the context of neurodegenerative diseases, neurons experience a lot of oxidative stress that seems to be driving disease. So what are these cellular pathways? Again, Raylin drove this work, and it's in a preprint right now. We addressed this in a genome-wide screen where we just looked in our neurons and stained them for levels of reactive oxygen species using a CellRox dye, and also for levels of peroxidized lipids, and fact sorted them based on high and low levels of those markers of oxidation and oxidative stress, and asked which genes either increase or decrease levels of ROS and peroxidized lipids. We found a lot of usual suspects that were known to play a role in this biology, but one hit stood out to us and really surprised us, and this was a lysosomal protein called prosapicin. Now, intriguingly, prosapicin has been linked to lysosomal storage disorders, maybe not surprisingly. It's a lysosomal protein. And actually, while we were working on this, another paper came out that actually linked it also to Parkinson's disease. We were able to validate the fact that knocking out or knocking down prosapicin, specifically in neurons, increased different markers of reactive oxygen species and peroxidized lipids. And intriguingly, this was very unique to neurons. It did not happen when we knocked down prosapicin in iPSCs or HEC293 cells, even though prosapicin is definitely expressed in those cell types. In other data, I don't have time to show you, it also didn't happen in other cell types that are pretty much post-mitotic in our cell culture system, astrocytes and microglia. So there was something neuron-specific that tied this lysosomal protein to reactive oxygen species and reactive oxygen stress. What's known about prosapicin? It's a proprotein. In the lysosome, it's cleaved by cathepsin D, a peptidase, into 4 saposin peptides, and they are very related to a 5th protein called GM2A, and these can act as adapters that basically can extract glycosphingolipids from membranes to feed them to hydrolases for degradation. And intriguingly, GM2A and cathepsin Ds were also hits in these screens, not as prominent as prosapicin or in both, but were also hits. So the first thing then to ask is, if we knock out prosapicin in our neurons, do we accumulate gangliosides? That was the case in a very dramatic manner. You can see we can stain for GM1, one of those gangliosides. It really is dramatically increased. And when we did untargeted lipidomics, we found, again, glycosphingolipids and polyunsaturated fatty acids also get strongly enriched when we knock out prosapicin in neurons. So far, so good, and that was kind of expected. We had a closer look at those neurons, and they showed a really striking lysosomal morphology. So normally these lysosomes under normal conditions are these relatively small punctate patterns. In prosapicin knockout neurons, lysosomes are engorged and have these big balls that almost fill the cytosol of the neurons. And together with Keshu at Berkeley, we did super resolution microscopy and saw that the GM1 is also accumulating in these engorged lysosomes. Are these actually functional lysosomes? Are we inducing kind of cells that are particularly good now in terms of lysosomal function? We looked at one important function of the lysosome, which is to participate in autophagy. And we found that actually these neurons, even though they strongly induce the autophagy components, they do not have an active autophagic flux. There's almost no flux. So basically, these are neurons that are maybe upregulating some of the components of the system, but don't have productive autophagy. So they're dysfunctional. What is the impact of having higher levels of reactive oxygen species? We asked this question to try to get further at this question of this connection between lysosome and reactive oxygen species. At baseline, these neurons are fine. You can knock out prosapis and nothing happens. But when we grow them in the absence of antioxidants that are a normal part of the growth medium for these neurons, while wild-type neurons are totally fine with that, prosaposin neurons at a specific point in time just all die, basically. And this was not through apoptosis. We can inhibit apoptosis and nothing changes. So what is the process by which they die? We did a modifier screen for survival of neurons, again, genome-wide in the presence and absence of antioxidants. Actually, most of the hits were the same in both conditions, but there were a few outliers that were knocked down only sensitizing neurons in the absence of antioxidants, not in the presence of antioxidants. And when we looked at them, the strongest one, GPX4, is a known inhibitor of ferroptosis, glutathione peroxidase. And intriguingly, the other three here are all in the pathway that generate the selenocysteine that's in the active site of GPX4. So that was cool. So that gave us the hint that maybe those neurons are dying by ferroptosis, which is a process where free iron leads to lipid peroxidation. And we can inhibit that by chelating iron or preventing this lipid peroxidation. In fact, that completely rescued the neurons. So knocking down this lysosomal protein drives ferroptosis. But how? So we did electron microscopy and found something very striking about these engorged lysosomes. They were extremely electron dense. And when you look at a textbook, this looked very much like lipofuscin, also called H pigment, which is a pigment that accumulates in old, typically post-mitotic tissues, such as neurons. It is autofluorescence, and indeed we went back, we saw this autofluorescence as well. And this was very cool. We're looking at young neurons. This is happening within 7 days of differentiation. It doesn't happen in iPSCs, it happens in neurons specifically, but we're recapitulating a feature of old tissues just by knocking out a single gene. And they accumulate iron, which we can stain for, and the iron co-localizes with these lysosomes. So based on some other literature, we have a new model then. By accumulating glycosphingolipids and also cholesterol, I didn't talk about that, we creating this kind of lipofuscin, these dysfunctional lysosomes that actually also trap iron. And they're no longer active in autophagy, but the iron trapped at low pH can actually promote the Fenton reaction that generates Ross species, which then can peroxidate lipids. And we showed that the polyunsaturated fatty acids thought to be important for ferroptosis also accumulate, driving neuronal ferroptosis. So with that, I just want to thank my wonderful lab who've been just amazing team players and helping each other out during the pandemic. Our funding sources, I have to give a big shout out to the Allen Institute for really supporting our early work to move into the iPSC space with an Allen Distinguished Investigator Award, and happy to take any questions.
Thank you, Martin. We have one question in the chat from Jim Rhodes. What happens to lysosomes in non-neuronal cells treated with PSAP?
Yeah, we don't see the morphological changes in them. What we haven't done is and we don't see the GM1A accumulation. And so I think part of the story is probably that neurons are very rich in these glycosphingolipids that are that rely on prosaposin for their degradation. And that could be one of the reasons that we see a neuron-specific effect here.
Thank you, Martin. There might be a couple of questions coming more through the Q&A. Our final speaker of the day is Dr. Ron Vale. Dr. Vale is a Howard Hughes Medical Institute Vice President and Executive Director of Genelia Research Campus. He studies the molecular motor proteins, kinesin and dynein, which power the movement of membranes and chromosomes along microtubules inside cells, and explores the roles of cytoskeleton in cell division and cell shape. In addition, Dr. Bill and his team employ biophysical and cell biological approaches to dissect the mechanism of T cell signaling and the mechanisms underlying neurodegenerative disease. Welcome, Ron.
Great, thank you so much, and thank you for the invitation. Let me share my screen with you. So today I will talk about another passion of mine, which is biology education. And I'd like to tell you about a project called Explorer's Guide to Biology that I've been working on recently. So anyway, just to step back and give you a big picture, we obviously invest an enormous amount in life science research. So this shows, for example, the US investment in biomedical research. But I also think that our scientific research has to be placed in context with the role of science in our society as well. So even in the past year, we've been seeing an increasing mistrust and misunderstanding of science. Even in the context of the recent vaccine with COVID-19. And even within our own scientific enterprise, this year has been a realization or a re-realization that, for example, representation of underrepresented minorities in science and the US population are greatly mismatched. And so I think we also have to consider all of these questions if we're going to have a society that will continue to support our research efforts. So I do believe that in addition to excellence in scientific research, we also have to think about excellence in science education and communication. And that this also needs to be a national and global mission of our profession, so that again, we have a well-educated society that understands science, can make rational decisions, and again, continue to support all the work that we do. And here, although as I just pointed out, there is a very large investment in scientific research, both in academics and obviously in biotech and pharma. I don't think there's a matching investment that's going on right now in education and communication, both investment in terms of dollars, but also in innovation as well. And I also think that this is something that our scientific community needs to think about. It needs to be involved with. Obviously, we have to engage in the research, but I think this aspect is something that we can't, in terms of education, communication, can't just outsource as well and expect it to be solved. This is an important issue that we need to embrace and think about as well. I think the encouraging signs are that, at least from my assessment, talking to many early career scientists, whether they're undergraduates or graduate students or postdocs, these are issues that young scientists actually want to be involved with, which I believe is very encouraging. That they're looking for not just completing their research project, but they're thinking about their lives and their careers and the profession, as something bigger, and what their roles are in society as well. So I do think there's an encouraging future, at least in terms of the ideas and perspectives of younger people entering the scientific profession. So in that spirit, I I also think that there's some very positive movements with regard to democratizing science communication as well. And the democratization should, really involves two things. We need to create very high quality content for science education. And that's an element also, I believe, of innovation. And I know the Allen Institute is also thinking very much on this line. And I applaud the Allen Institute for all the great work they're doing in terms of visualization, which is a great tool for science education. And the democratization also has to be accompanied by making all of this great content freely available. And I think we're seeing this movement to open access right now for scientific literature, for research articles. I think this movement needs to be extended to educational materials as well. So anyway, these are issues that I've been, well, I must admit, early in my career, all I thought about was science and scientific research, and not very much in teaching either. So, probably later in my career, I began to realize that these are very important issues. And I think, again, the encouraging news is that we can mobilize the scientific community for these kind of communication efforts. So this is just one example, I would say, of an effort to rethink the traditional seminar. Obviously, the traditional seminar is flying people, which we're not doing right now, but from one place to the other to give the seminar. And in this case, it's often kind of elite scientists flying to other elite institutions to give their talk. And in 2005, I made a trip around, this was pre-Zoom, around the globe to India, and realized I traveled halfway around the world to speak to about 150 individuals at a very elite institution in India, and just realizing that there's so many people that don't have access to seminars in smaller institutions in many places around the world, that either can't afford it or can't attract elite speakers. After that trip, with about $2,000, this project got started. It was originally called iBioseminars, now iBiology. I think many of you know it, but it has been an effort since 2006 to film, all kinds of scientists and put it on this online environment so anyone around the world can view it. And this project continues. There's a fantastic team there now led by Sarah Goodwin and Elliot Kirchner, Shannon Berman, and others who are working on this project. So more recently, I've also become involved in this other project, which is an effort to rethink the introductory biology textbook. And the reasons are, first of all, they're expensive. And in fact, so expensive that often at $250 or $300, this is just prohibitive to many students who are taking biology around the US, even in some of our richest areas, such as Seattle or the Bay Area. The second thing, I don't think that these books are really doing the job of taking someone who's being introduced to sciences or biology and exciting them about the subject so they want to learn more. In many cases, I think even among scientists, we don't eagerly pick up one of these books for an exciting read. So if we're not excited about reading them, it's kind of a hard ask to get young people excited in the subject matter through this kind of content. And I think the other shortcoming is that they're basically a sea of information, and that's kind of largely how students see biology. which is unfortunate. And there's very little insight into how new knowledge is derived. So really, this project emerged again as an idea, a retreat actually in Princeton with a bunch of scientists and artists and filmmakers, no money at a time. And out of this launched kind of at the end of last year, the Explorer's Guide to Biology. So it's now a website, it has some money, it's still early on in its journey, and it has a director now, Mike Walter. So at least it started. I'll just give you a few ideas of what makes it different from a regular textbook. First of all, it's free. And, you know, I think this is going to be an enduring, mantra of XBio, that it will be free to the end users, which are students and teachers. An effort to make it engaging. And I think very importantly, really presenting science. And what science is not information. Science is a way of knowing the world. It's a way of uncovering new knowledge. And, you know, that's what I think we want majors or non-majors in biology to understand. The strategies are different. A traditional textbook has a few authors and a publisher. And I think what we're trying to do here over time is really make XBio a project of our entire scientific community. Textbooks lay out a defined order of chapters. Here we're trying to make anyone can pick up any content piece in XBio and read it. And there's some strategies there that I can discuss. But the intent is not a fixed order, that you can pick up whatever you want to read and use it in a modular manner. Textbook takes years to revise. XBio is online and more nimble. And the goals are really to understand scientific thinking, really, and that means how to ask questions, how to evaluate evidence. That's the core of science. Yes, students need to learn key concepts and information, but I think we're just giving them far too much information right now. And I think there's some skill in actually making it more selective and less encyclopedic, what we want to pass on. And this is also an important goal, I think. I have a love of literature right now, and I trace my love of literature to the English courses that I took in high school and as a beginning undergraduate. And I think these courses have done a spectacular job of explaining the craft of writing and to appreciate good literature. And I think we should try to aim for similar goals in science education as well. And I think we shouldn't shy away from this beautiful quote from Marie Curie, that in science, I'm among those who think that science has great beauty. I think that many of us who've made it now on this Zoom meeting to become scientists believe this is true, and that's how we got inspired and why we love our profession. And I think we need to pass this on to other people that can, appreciate science as something really creative, interesting, and that does have beauty. And I think with XBio, we're trying to do this by through a storytelling approach, that there's humanity and great stories that are involved with every discovery, through more accessible type of art rather than a lot of our standard drawings in textbooks, and through videos that allow students to actually meet scientists who made a number of discoveries. So, I think it's continuing to grow and evolve. I encourage you to look at the website. Do I have a couple minutes left, or am I pretty much at the final moments here?
You can go a couple more minutes.
Okay. Well, in that, maybe I will give you a 2-minute view of the website right here. We have contents in genetics and cell biology, a bunch of material for educators. This is our initial group of authors. We're going to grow and also create more diversity in this author list. But there are a number great people, including Nobel Prize winners who have contributed, I should say, without even any honorarium, because they believe in this project. And since Martin talked about CRISPR, we can go to this one example of CRISPR by, Jennifer Doudna, this year's Nobel Prize. And this is an effort to really, in this case, it's called a key experiment, to describe, what is it that led to that Nobel Prize? And I don't have time to go through the whole thing, but to give you a general idea, in this case, there is one key experiment. It's one panel in this paper that really changed the world. And so this is a piece of data, and, backing up, the goal is really for a student to understand what this data is. It involved control experiments and one real experiment on CRISPR cutting, but it goes all the way from describing what a gel is, how to run a gel. This is a video in a virtual classroom. It explains, what the experiment actually was, but it always, all these things begin, I should say, with a story. So it's a really personal narrative of, what happened in this journey to discovery here. And it also, describes some of the humanity of science, that they had no idea where this was going, that it would lead to genome editing, and also the humility that some of the initial experiments that they did fail. And I think that's also something important for students to understand. So I encourage you to, look at the site for additional information. But, again, there are other ways I hope to engage with the Allen Institute to help build this community goal as well. I've already talked to Graham Johnson on several occasions about it. And with that, I'm happy to answer any questions.
Thank you, Ron. The storytelling of science is very beautiful. Okay, so while we wait for questions to come in later, feel free to answer the questions in the Q&A panel afterwards. Thank you so much for the presentation, Ron. To conclude today's session, we're having a moderation on a panel discussion with all speakers from the day. Members of the Symposium Committee crafted these questions to discuss the speakers' interests in challenges within and the future of cell biology. We're going to ask all speakers to join us on camera now, and I will introduce our moderator, Brock Roberts. Brock is a scientist on the stem cells and gene editing team at the Allen Institute for Cell Science. Take it away, Brock.
Okay, thank you, Calista, and thank you to all of our speakers for the profound, stimulating, and interesting talks. It's really an honor to follow that last talk. Thank you so much for what you said, Ron. And we have some questions that we've made up and that we hope will stimulate a really interesting panel discussion. So I think I'll just jump right into them. And we have sort of designated subsets of the panel participants for each question. So I'll mention who those individuals are at the beginning of the question, so those people can pipe up, and then I'll ask the question. So the first one is for Derek and Susan. Why don't you guys just answer in that order, please, for no particular reason, but just to introduce some organization. And the question is, while cells can be studied in isolated systems, tissues are more realistic representations of the natural environment that cells exist in. What tools or methods do you think will help us study complex interactions that cells might have in their natural environments?
Well, that's a rough question given that I'm a cell biologist that studies strictly cells in 2D. But that being said, I think one of the reasons why I've tried to incorporate Drosophila into my research is that I am allowed to, I can go from looking at things with high resolution in cells and then start to look at some of those questions inside the organism. And so, you know, Drosophila, I think, are a great model. I don't think I need to argue that with anyone because there's so many different reasons why. But I really do like the interplay of looking at things in 2D and in dishes and seeing really high resolution imaging and getting details that you can't necessarily get from tissues. And then looking at specific processes inside the cell. So I'm a big fan of Drosophila. I like the interplay between using cell biology in dishes and using cell biology in organisms to sort of answer questions. I'm not sure if that answers the question, but that's kind of my ideal approach.
Okay, yeah. Oh, we have a muted microphone. Susan, please. Sorry.
Sorry.
Can you restart?
Yeah, I agree. I mean, I'm biased obviously because we work a lot on single cells, and I think it's important to work on single cells as well as in the context of tissues. I mean, with the revolution of things like cryo-ET, super resolution microscopy, you can do a lot of things in situ now that you couldn't do before. So you can see what a cell is doing in the context of its environment. But looking at individual cells for the sake of themselves simplifies the system and lets you actually find a lot of the mechanisms that you can't find when they're in situ because there's too many interactions going on. And I think you need a really good balance of both of those.
Okay, yeah, and balance is, I think we had some good examples of how to achieve that balance in your talks. Thank you. And the next question we have is for Martin. And that question is the following. New techniques are giving us tools to study single cell behavior in addition to the more traditional population-based study of cellular behavior. What new information do you think we'll be able to extract by looking at single cell behavior rather than population dynamics?
Yeah, I mean, I think there's already great examples for rare cells that can actually be really important if you think about processes such as cancer, where sometimes a super rare population or the immune system, right, a super rare population of cells can be functionally really important. And that information could get lost if we just average out properties over whole cell populations. In my area of research, something I'm really fascinated by is the selective vulnerability of specific cell types, for example, specific neurons to disease. And again, this could sometimes be a needle in a haystack, where if you want to really understand how the disease first starts and what are the factors that make a cell vulnerable to disease, you need to be able to really look at those rare populations and those specific cell types.
Okay, thank you. Yeah, that's incredibly interesting. And we have a question next for David. The question is, machine learning has clearly been a key technique in being able to analyze large data sets, whether those be image-based or other data sets. However, extensive training and large data sets are often needed to implement these approaches. How do you think we can democratize the use of these advanced image analysis techniques?
Yeah, that's a very good question. Yeah, so personally, I think like democratization is not going to be possible without a massive public investment in a new data infrastructure for the life sciences. The reality is that these are for many of these things, for many of the things that we want to be able to do. You sort of need like that level of support, and it's hard to imagine a world where everybody gets to partake in the artificial intelligence revolution without having like a public, well-supported, well-developed infrastructure for supporting these larger data science driven projects. So in absent of that, like I don't view this as being like democratized. What will likely end up happening is that the labs that have sort of either the specialized expertise or specific interest, they'll generate the data sets that solve their problems, and then the expertise and the data remain siloed. With a larger infrastructure, I think it'll be possible to actually have these methods like available for everybody. But it's going to require a substantial change in the way science is done and the way science is funded.
Mm-hmm. Okay. Next question we have is for Martin and David. This is a little bit more open-ended, a little bit more philosophical. I hope that's okay. There are many interesting things going on in this field. How did you identify the thing that is most interesting to you? And let's do Martin and then David, please.
Yeah, that's the question, right? That we all face. Like, what do we want to spend our energy and time on? I actually, when I started my own lab, I was actually trying to not think about what's going on in the field, because that means a lot of people are already working on it, but rather to maybe return to some of the really, really old questions, that we still don't know the answer to. For example, we've known for more than 100 years that protein aggregates are correlated with neurodegenerative diseases, right, in the brain, but nobody knows how they act. Why are they actually bad? I mean, there's still a huge unanswered question. And so I think sometimes it's worth not to think about what the field is doing, but really to say what's a really old question that's been fallen by the wayside a little bit. And do we now actually have fresh tools to address that old question?
Okay.
Yeah, I would say for myself, I have a little bit more of a Larry David approach to science. Like I cast a relatively wide net, and then there end up invariably being some things I just can't let go. And those end up being the things I end up working on. Sometimes it's a classic system that, I think is like really, really elegant. Sometimes it's a piece of technology where it'd be like, man, it'd be really cool if we could make that. Sometimes it's something, in this case, it's like, we want an algorithm to be able to do it and to do something. And, what is currently out there just isn't able to do what we want it to do. And you just stick with it. So yeah, I'd say that's kind of like how I view things. It's, many things are interesting, but some problems just tend to stick around in the brain longer than others. And those are the ones that end up taking the most of my time.
Can you identify Larry David for those not aware?
Larry David was the creator of a number of TV shows. So, Seinfeld is probably his most popular one, and Curb Your Enthusiasm is another very popular one. And, we're, at least in my household, we're going through some of the older Curb Your Enthusiasm episodes. And so it's kind of an analogy that made sense.
I appreciate that response, and I'm a huge Seinfeld fan, but I'm also aware that there are many scientists shaking the field all the time. There's probably one named Larry David that I may have.
Yeah, I would say that. So in the show, like this, like fixation is not like a positive personality trait for most of the episodes. Like I'm personally asking, like, if you just let this thing go, then there'll be like no conflict, and the episode will like nicely resolve itself. But it never does. Although for science, I think it's actually can be a positive trait.
That's wonderful. Okay, we have a question for Ron now. Ron, where do you think are the what do you think are the exciting directions for the field of cell biology to take, and what challenges are interesting? How might collaborations help us make more progress in understanding these questions?
That's a pretty all-encompassing question, which probably has many answers. I mean, I could just tell you at Genelia that we recently kind of announced kind of a new direction for a research area where over the next several years we'll be hiring 15 group leaders. And that research area is called 4D cellular physiology. And really the spirit of that is to move cell biology more from reductionist systems. And what I mean by reductionist systems is either cells in culture or biochemistry, and develop more tools for studying cells in the context of tissues, living organs. Obviously, the neurobiologists have been doing this for years because they've had to study neurons in the brain. All the connections couldn't be reduced to a cell culture environment. But, I think they're all many fascinating questions of how cells are communicating in tissues and working together, to create physiological organ functions that we don't understand. So I think that is one exciting frontier of cell biology and kind of an exciting direction even for my own lab. I've been a reductionist cell biologist for my entire career, and this will be a new challenge even for my own lab as well, but I'm looking forward to embracing it.
Wonderful. Okay, yeah, we're really excited what happens to see the future at Janelia. Next question is going to be for all the panelists, except for me. And I'm just going to go clockwise from how it appears on my screen. So that'll be Martin, David, Susan, Derek, and Ron. And the question is, how do you envision different layers of information being transmitted across different scales? For example, how can we connect molecular information to cell information to tissue information?
Sounds like Ron has a plan for that.
Hey, no, I'm last. I want to hear what everyone else.
Yeah.
No, I mean with Janelia, right? I do agree. That's a great question, because I do think that's kind of the cutting edge, right? To kind of start to connect everything through from the molecular, a molecular level to cell, to tissues, to organismal level things. And I mean, we've heard some great talks today about some of those things from Derek and others, right? So I think that is a really exciting new area. And yeah, I think we need to have the right biological systems that allow us to study that.
Okay.
That is a good question. I would say my approach would likely be some form of knowledge graphs, and having making sure that the experiments one did focused on actually building those connections across different those different like length scales, from the molecular to the cellular to the tissue level. I think knowledge graphs would likely be a good start, because there's vast amounts of information at each layer, and I think the experts in one layer are often unaware of what's happening at the other layers, and sort of a unified effort to assemble the knowledge, sort of coherently explain like what's known, but also like what's unknown. I think that's kind of like what I envision as the first step of that kind of process.
I guess one of the first things that occurred to me is that there's a sort of a practical issue here, because when you try and extrapolate what you learn in one system to others, sometimes it doesn't work very well because there's differences in those systems. So it might be good to find molecular models that allow you to go from, say, a cell to a tissue to an organ or whatever, and be able to look at that in addition to looking at separate ones, so that you can extrapolate and not lose a lot of information.
And I would say, Martin showed this beautifully. Different cell types have different, effects when you knock down proteins. And I think my approach would be to think about cross-correlating different cell types and looking at different phenotypes, and that way you could sort of get a sort of a holistic picture of what's happening by cross-referencing different expression levels in different cell types, and kind of using the variety of cells that are out there to start to think about how this one particular protein, and it's mutated in this way, one way, might affect different cellular processes.
Okay, and then lastly, Ron, please.
Well, I think what you're hearing is probably none of us have the answer, and I think I think this is really the challenge of our quote unquote generation, is to work on and think of that problem. And I think because we don't have the obvious roadmap of how to do that right now, it's going to take a lot of creative thinking, and I think part of the route to that is, really getting interdisciplinary communities thinking about it from different perspectives. So, like I was excited to hear, David's talk and about, bringing machine learning into data sets that have been traditionally domains of cell biology. That's a whole new exciting direction that it remains to be explored. How to integrate the data sets that Martin was presenting of, genomic data with, other kinds of data sets like the cell images that David was showing. I mean, how we kind of integrate, these different qualities of data and make sense of them together, I think is a great upcoming challenge. And there are all these epics of biology where we look at biology and there were always new tools or new ideas, and we couldn't imagine the path forward, and then bright people develop new technologies or new conceptual ways of thinking about things. And so I don't know, I think this is the question of our time right now, and be exciting to get lots of people thinking about it.
Wonderful. Yeah. Okay, great. We have actually some really insightful questions inspired by the last talk, Ron's talk, centering around education. So I'd like to toss this question first to Ron, and then open it up for the other panelists to answer if you like. And the question is, how do you propose creating incentives for teaching innovations by elite early scientists when the current funding structures and tenure/hiring mechanisms rely so heavily on research, and inevitably focused time on teaching is just time judged to be not spent on research?
Should I start with that?
Yes, if you don't mind. If you could start, and then have others
hear
chime
everyone's
in.
thoughts on that.
If you want to, you can kick it out. If you want to phone a friend, that's fine.
Okay, I'll pass it on to Derek. Derek's at Reed College, so that's like one. I would imagine there's more incentive, that would be a high incentive there. But how does Reed, which has a fantastic reputation in this, incentivize innovation and best practices in education?
Yeah, so I'm gonna geek out for a second. Ron is one of my biggest influences in my entire scientific career, and I have kind of always thought and looked to him as someone I'd like to emulate in my own life. So it's kind of weird for me to be on this panel because this is one of my heroes. Anyway, I think Reed's ethos has been teaching as research is teaching. And so I think, and I also take pause with the word elite and being at big institutions, because I don't think that elite scientists are just at big institutions. I'm not saying I'm an elite scientist, but I think there are a lot of great elite scientists at, the word elite, at small institutions. I think incorporating research that you're doing that is peer reviewed and out there in the world, and bring that into the classroom is a way you keep fresh and be competitive and train students that are ready for the next generation. So I think as much as we can incorporate inquiry-based real research into our teaching and have your teaching blend in with your research, that's how you can stay competitive and can train the next generation to be ready for whatever is out there to come.
Okay. Do we have other panelists that would like to chime in on that?
Yeah, I would completely agree with that. I think I think project-based learning is just wonderful for students, right? And being exposed to seeing real actual research and having a part of that, I think is something that is hopefully something that can square these two things, exactly like Derek said. So I think that could be a great way forward, I think, to broaden project-based classrooms that actually engage students in ongoing actual research projects.
Okay. And Ron, we have another question targeted for you, also in this realm of education. This actually comes from Cynthia Fisher. And the question is that in curating educational resources, how should an educator or publisher approach quality control and accuracy of information on this platform, as opposed to how that process is managed in writing a standard intro biology textbook? And we would also welcome input from other panelists.
Yeah, I think pointing out a very important thing that, crowdsourcing is a very powerful way of engaging the community. And, it's a chance for involvement and I would like to grow XBio as a way where many people could contribute to it. That said, there has to be, central quality control. So, I mean, at one level, if we ask someone like Jennifer Doudna to write a chapter on CRISPR, there's a good chance that there's going to be good quality control in what comes back. Or Shirley Tillman to write about Mendel because she knows genetics pretty well. But looking in the future, one could imagine, 60 chapters covering all of biology written, by various people who are experts in these subjects. So, that's how you get very good quality control. It doesn't, in fact, maybe better. It's a challenge actually for 3 authors to write about all of biology, in many cases in spheres that they don't know that well.
Yeah.
So it's also, the notion that everything in a standard biology textbook written by 3 people has uniform quality control throughout the book is not necessarily true. The other thing I'll generally say is, although right now we've asked a lot of, quote unquote, well-known scientists to participate in the project, kind of the next stage is we'd love to engage the educator community as well. Because there are, Derek, an example, there are, fantastic educators who, teach undergraduate education and place high value in that and are really good at it. And we'd like to get that community involved in the project too. So I think to some extent, in answer to your question is, I think we'd like to engage many people, but part of the quality control will be engaging many leaders in the scientific and educational communities.
Wonderful. We have a question. Did anybody else want to answer that question? I would like to make that opportunity available.
I was just going to say, textbooks are wrong all the time. So that's not new. And also science is a process and not a collection of facts, and it's self-correcting. So, maybe the quality isn't great, but I think in the end it will shake itself out because it's what always happens.
Mm-hmm. Other responses? Okay. We have a question about kind of current times in the context of science. Our question comes from Harold, who is curious to hear about the thoughts from each panelist with regard to how you have observed the unfortunate growing mistrust of science during your career. Why don't we start with that? How has this process of mistrust been observed from your point of view? And we'll just call on Martin, David, Susan, Derek, and Ron in that order. And we can do phone a friend.
Yeah, I hear about mistrust from science a lot through the media, of course. I guess I'm quite fortunate not to have too much interaction myself with that, having been confronted with that. But yeah, I do think it's a really serious issue that is a high priority for all of us to address.
That is a very serious issue. And I think there are, I would say that the cases that come to mind sort of make me wonder whether the scientific community has actually done a good enough job actually being trustworthy. And so so I'd say the immediate example of this is actually vaccination, right? We're in the midst of a global pandemic. We're fortunate that thanks to some brilliant scientists, we have vaccines that are coming along the way, but there are communities that have just been horribly mistreated by both the medical industrial complex and also the scientific community that just don't trust that these vaccines will work, and they don't trust that these vaccines will actually be beneficial to them. And that mistrust is rooted in like very real, you know, harmful, almost genocidal experiments that, you know, I don't want to generalize, but like this community of people, science at large, has done on those populations. And I think it doesn't make, like, when I hear people talk like, oh, like, how can people not trust science? And it's just like, it's very easy if, you know, 3 generations ago, like they were experimenting on your grandmother to try to like perfect their surgeries. So I think like we actually, you know, as a community, like scientists just need to acknowledge that, you know, we're not the most trustworthy. You know, the things that we do end up reshaping society in drastic ways. Oftentimes that reshaping is not beneficial for the people who have to live in that society. And, you know, own up to that. And also I'd say, you know, in light of that, actually, you know, make sure that, you know, the vast majority of what we do is for the benefit of people. And I feel if we do that, then it'll be easier to make the case that, you know, science is in and of itself trustworthy. I'd say like I have a, you know, I sort of have like a relatively like unique position because I'm working in a space sort of at the intersection of artificial intelligence and, you know, cell biology. You know, the algorithms that we make, as you see today, like they're going to lead to, you know, some really exciting advances in tissue biology, as well as, you know, advancing like different, you know, therapeutic programs like across the country. But the reality is, you know, you take artificial intelligence and you look at like what it's done as far as, you know, policing, facial recognition, recidivism recognition, and like it's a crime against humanity that shocks the conscience. And when you realize that this is what's happening to like those communities, who honestly, like many of them don't even have a voice, you know, it's very easy to realize, to see like how, you know, we're not to be trusted.
Mm-hmm.
So I'll get off my soapbox.
Wow. Yeah, no, thank you for your response.
Yeah, this is a tough question. I mean, we can certainly all do this in our local community by talking to people and explaining things that they don't understand. But it's really disturbing, I guess, when you see national figures like Dr. Fauci out there trying to help people and they get crucified in the media. So I don't know how we deal with that problem very well, because, I mean, if you have a public platform and that's not working, I'm not sure what else you can do except to keep trying.
I second everything David just said. I think there is a history of abusing populations. This is not a new thing, right? The thing I will say though, demystifying science is important, and this is a plug for liberal arts. I think lawyers should know science. I think teachers of other subjects should know science. I think everybody in society should understand and have an understanding of science. And one thing that I've been really impressed at working at the Bark School is that every student that comes through Reed College has to have some sort of science training or some sort of class. And hopefully what they take away from that is at least a basic understanding of science. And when science is in through all parts of society, not just in the scientists, it's not siloed, I think we get a better chance of people being more trusting of it.
Okay, yeah, finally, Ron.
Yeah, I can be brief. I mean, I think a lot of the comments that have already been made are really exactly to the point. I mean, the other thing is just to say, finding more ways to engage people directly, you know, in conversation between the public and scientists. So just as an example, what we now call the Wonder Collaborative, which is a branch of iBiology, and that was spearheaded by Elliot Kirshner and Sarah Goodwin, produced this film called Human Nature, which was kind of an effort to get ahead of the game with CRISPR. So I think also if you lose the narrative early, which has happened in many cases, for example, with vaccines being an example, or with GMOs, if you lose the narrative early, and because the scientific community never made an effort to get accurate information out, it's very hard to recover from it subsequently. So I think the community has to be a little more, you know, proactive in the communication and discussion. So on the positive side, so Human Nature is a film. It's available on Netflix now and it just appeared on Nova. was a film to kind of explain what CRISPR is, what the discovery is, how CRISPR works, but also some of the ethical issues that are being debated right now that are, difficult, and that's for scientists as well to really grapple with how to use the technology properly. On the encouraging side, when that film has been shown in public settings, and there has been a Q&A discussion with the public and scientists afterwards, it's been fantastic and very engaging, and the public has appreciated it. So I think there are ways of bringing difficult and, legitimately complex issues for discussion between science and the public. And I think we just have to find more venues in which we can do that. And I mean, to some extent, I think we haven't tried hard enough, quite frankly. And therefore, always being an optimist, I think, I think there's an opportunity, for shifting the trend where we are right now. But it does mean that our community, it has to be, again, be one of the priorities of what we do, again, as a scientific community.
Thank you all for your answers to that question. We have a couple quick hitters to kind of finish up on. And I think I'll scramble the order, make it alphabetical by first name. So that'll be David, Derek, Martin, Ron, and Susan. And that question is, please tell us about the biggest surprise that you've encountered in your career.
The biggest surprise, yeah, I would say, I mean, I'm not sure if I'd call it like necessarily the biggest, but a very nice surprise has just been that, realizing like you don't have to be perfect to be a scientist, and that, sometimes you can, make errors, things can be incorrect, and, as long as you acknowledge that, and of course you're being supervised, being forthcoming, but that things will ultimately end up being okay. I think the undergraduate, the flavor of our undergraduate education system sort of encourages people to have a mindset that you never make mistakes. Everything has to be perfect. You need to get A's in absolutely everything. And I think sort of leaving that mindset and being sort of realizing like, hey, you can actually explore in areas you're less comfortable and make mistakes, and that's not going to tank your career. I think that was like a very nice surprise.
Okay.
Yeah.
Wonderful.
I think the biggest surprise is the strength of friends, like the network that you can build, and how kind and caring and willing that people are to give time to you. I think I've always spent my time afraid to take up space, I guess, and ask people questions for help or for reagents or anything like that. And how kind and giving, I guess, the people that I've asked has been the biggest surprise to me. And I kind of kick myself for not being a little bit more bold and being to ask people for help and for reagents, and for just like, will you listen? I have a grant idea. Do you mind talking about it? And people have been so willing and kind to do that. That surprises me, because I guess I've been sitting there thinking that people didn't want to be that open and collaborative. So, but maybe that's just the people I've asked.
Okay.
For me, one very nice surprise has always been, since starting my own lab, that I see really my job a little bit like a coach, but that I actually, my main job, I feel, is to create an environment where people do their own thing. And I love the surprises that people bring to me when they have their own ideas, discover something, come to me. And it's maybe something as a postdoc, you're much more kind of like down in the weeds and in control of every little thing. So as a PI, to step back and let people in your lab surprise you with what they did by themselves, I think that's a really nice aspect of my work.
Okay, and for we'll move next to Ron.
Well, I would say the biggest surprise is that science is a surprise. And what I mean by that is that universally discoveries always come about from surprise, I would argue. You know, because one way that people think about science is, again, a misconception, and maybe that takes a long time to realize, but you know, that you have this idea in your mind and then you go out and prove that your idea is right. And of course, we often do that, but you know, I think the key for being a scientist is to look out for the surprise where your idea may not be right, or you know, a little clue that there may be some other path that no one has thought about, and that's what discovery is. So I would say, you know, scientific discoveries always emerge from that, and that's what actually makes science fun and interesting, unpredictable, and a joy.
Wonderful. Yeah. Okay, we have a final question. Why don't we go in reverse alphabetical order by first name?
You
Dr.
skipped
Miss Susan.
We didn't get Susan. Yeah.
Sorry about that. I was jumping ahead. Susan, please.
For me, I think it's how many times you can be surprised by people's results. I've been to a great number of meetings where you're sitting there and all of a sudden somebody shows something that is just so beautiful, and you think that, you know, you've seen it all at some point, but that just doesn't happen. And you walk away thinking, wow, I wish I'd really done that.
Apologies again.
It's okay.
For our final question, I'm thinking we'll do reverse alphabetical by first name. So that'll be Susan, Ron, Martin, Derek, David. And our question is, 25 years from now, the 3 transformative technologies in cell biology will be stem cell biology, CRISPR, and blank. And if you would like to challenge either of the technologies that we've placed on the podium, that's fine as well, but please justify your answer.
I guess it depends if I'm looking at it from a cellular or molecular point of view. From a cellular point of view, I think it's super resolution microscopy and all of the ability to see almost individual molecules at this point. If it's molecular, it's single cell omics, because no matter which kind of omics you do, being able to look at thousands and thousands of cells and bring it all together is going to make a huge difference.
Okay. Okay, and then we'll reverse it to Ron.
Well, I'm the oldest person here, so you know, it's questionable whether I'll actually have to worry about figuring out whether that prediction is true or not. But anyway, I'll try. Anyway, I don't know. It's a great question. You know, listening to David's talk, I do think that you know, machine learning of thinking about very, very complex pieces of information that human beings have a hard time digesting may not replace us as scientists, but provide a very powerful companion. So I would put that, even though I'm not an expert in that space, I'll put that in the 3rd category.
Okay.
All right. Well, I like the first 2 categories because we're working on that. Although I bet in 25 years, there's going to be completely other things that are cutting edge, you know, and it's hard to guess. I would think that that really, I think, echoing Susan, I think molecular imaging, really looking in a cell where all the molecules are, whether that's by super resolution, spatial transcriptomics, proteomics, you know, CLEM, fib milling, but, you know, integrating a lot of these technologies to ultimately really see where all the molecules are, ideally in a live cell. I think, you know, 25 years, a lot can happen. So I think that would be very exciting.
Okay.
I'm going to take a cop out and actually take a bigger step back and just say the breaking down of the walls between disciplines, which I know has been happening, but I think it's going to happen more and more. And I mean, I absolutely love having physics students in my lab, and I love watching my ecology colleagues bring in, you know, data scientists and statistics and modeling into their work. And so where all these disciplines of science used to have their own little silo, I'd love to see all of them coming down and everything just being really mixed up. And we're all just scientists studying a process and trying to figure out an answer and then doing whatever it is to find that answer. I think breaking down those walls and being more integrative will be the next thing.
Not a cop-out.
Yeah.
Yeah, please, David, finish us.
So I'm tempted to agree with Ron and say that I think like machine learning and artificial intelligence, like those methods are just have immense potential for helping us make sense of really complicated data sets. But I think the reality I feel it's just going to be vaccines. The reality is like this is like our third novel coronavirus in the past 20 years. We're likely to have more, and the ability to create vaccines on the time scale of like literally less than 6 months is going to make it so that we actually can keep pursuing our passion, which is cell biology. So I think that kind of needs to be acknowledged given the historic times that we're in. So indeed. I'm going to say vaccines.
Yep. We are 1 minute past the noon hour on Pacific Coast time. And I want to thank all of our participants for both their talks, but also their willingness to participate in this discussion. That applies not only to today, but to each of our sessions. So thank you so much. And the next stage in programming will be handled through closing remarks.
Right, thank you, Brock. Also, big thank you to all our speakers today for your excellent presentations and engaging discussion on the panel. Thank you, everyone, for joining us at the Seattle Cell Science Symposium and for your engaging questions. This concludes the webinar for today. For those of you who are registered to attend the Cell Science Tools Q&A session, we will see you in that separate Zoom meeting at 12:15 p.m. PST. If you have questions about the 4 allencell.org resources highlighted today, you can find the Zoom link for the Q&A session via the link in the chat. As always, we also wish to thank our founder, Paul G. Allen, for his vision, encouragement, and support. Have a wonderful rest of your day. Thank you.
Thank you.